Europe, and good afternoon. Oops, sorry. Good afternoon, everyone in Australia. So welcome. My name is Wojtek Szanowski, and I'm a Deputy Director for Academic Partnership at Sydney Nano Institute. And we have a great pleasure today to host, together with IIT in Italy, a Bio-Nano International Workshop. Before we move to formal proceedings, I would like to acknowledge traditional owners of the land on which University of Sydney is located and built, the Gadigal people and the Eora Nation, and we pay our respect to the knowledge embedded forever within the Aboriginal custodianship of the country. So, without further ado, I would like now invite GIANNI, who will be presenting as the first, to his presentation on piezoelectric materials in cancer nanomedicine. Just a reminder, this meeting will be recorded, so all the recordings will be then posted on a YouTube channel. If someone would like to ask a question, feel free to do it through the chat function, but the preferred option would be if you, at the end of the presentation, unmute yourself and ask the question directly. So now, over to you, GIANNI.
Thank you very much, Wojtek, for the introduction and for the organization of this workshop. I am just trying to share my screen. Yeah. Can you see my presentation? Okay, perfect.
If you could put it, please, on a full screen.
Yes. Just a moment. I was checking that everything was working properly. And so thank you again. Good afternoon there in Australia. Good morning to everyone here in Italy. Actually, at the last point, I decided to slightly modify the content of my presentation, and I apologize for this. But I would have preferred to give a whole overview of the activities that we are performing in our lab, despite instead of focusing just on piezoelectric nanomaterials. However, piezoelectric nanomaterials will be a part of this presentation today. So this is IIT network, Istituto Italiano di Tecnologia, that as probably you know, is a quite young institute operative since 2005. There is a main headquarter located in Genova, and then there are several other centers that are spread through all the country. We are located in Pontedera, as I was mentioned before the workshop, is a little town close to Pisa, most famous for the Piaggio factory and the Vespa motorbike. And in Pontedera, I am the coordinator of the IIT Center, that is the Center for Materials Interfaces. At the Center for Materials Interfaces, I am the principal investigator of the Smart Biointerfaces research line. This is a very recent photo of our group. We catch it 2 days ago. And these are our main supporters in terms of projects and the grants, in particular the European Research Council and the Italian Association for Cancer Research. So in our laboratory, we are interested, of course, in nanotechnology, nanomedicine, and specifically, we are trying to exploit the active physical properties of nanomaterials and nanostructured materials in order to obtain and produce active nanovectors, active nanoparticles that can be externally stimulated in order to foster some specific functions inside the cells, inside the tissues. So rather than a traditional passive nanocarrier that is delivering a therapeutic agent or diagnostic agent, we would like to obtain an actual, let's say, nanotransducer that is able also to give a physical stimulus to the cells. These are the four main classes of nanoparticles that we are interested on. As I said, piezoelectric nanoparticles, so nanoparticles that are able to transduce mechanical energy into electrical energy and vice versa. Antioxidant nanoparticles that are sensitive to pH values and can behave as reactive oxygen species scavengers in appropriate physiological condition. We are interested in nanoparticles that are responsive to magnetic field in order to perform hyperthermia or in order to perform magnetic drug targeting. And we are interested in nanomaterials that are responsive to light and in particular to near-infrared radiation. So let's start with piezoelectric nanoparticles. Let's say that we pioneered the application of piezoelectric nanoparticles in biomedicine. We started, let's say, 10 years ago by testing the possibility to exploit very, very little nanometric piezoelectric nanocrystals in order to perform, let's say, a wireless stimulation of cells and of tissues. The idea is to exploit the nanoparticle as a nanotransducer, to incubate cells with these nanoparticles, and then to perform a non-invasive mechanical stimulation, for example, assisted by ultrasounds, and convert this mechanical energy into electrical energy that can activate the cells. So we demonstrated the possibility to perform such wireless stimulation in neurons, in muscle cells for application that are ranging from neural stimulation to tissue engineering, for example, for innovative cochlear prosthesis. And very recently, we also investigated the possibility to apply piezoelectric nanostimulators for cancer therapy. And this is a very recent study that we published a few months ago. In this study, we developed polymeric piezoelectric nanoparticles. Up to now, most of the piezoelectric nanotransducers are inorganic, based, for example, on zinc oxide, barium titanate, or boron nitride. But of course, despite their high efficiency in terms of piezoelectric properties, they can raise some concerns in terms of biocompatibility, in terms of long biodistribution fate in the organisms, and so on. So we preferred slowly to move towards fully organic and biodegradable piezoelectric nanoparticles. We lose something in terms of piezoelectric properties, but indeed we can exploit these biocompatible nanoparticles in a more safe manner. So this example is represented by a hybrid nanoparticles, the core of which is represented by a polymer, polyvinylidene-fluoroethylene, PVDF-TRFE, that is a well-known piezoelectric polymer. And then we have a lipid shell of DSPA PEG that is giving higher stability and high biocompatibility. At the same time, as we are intending to exploit these nanovectors for cancer therapy, we are also loading a chemotherapy drug in the inner core of the nanoparticles, and in this case, nuclein-free A, that is a non-genotoxic chemotherapy agent. The surface of the nanoparticles was functionalized with the ApoE, that is a short peptide that is promoting the blood-brain barrier crossing and the targeting of cancer cells, because maybe I forgot to mention, our main goal is the treatment of brain cancers, in particular glioblastoma multiforme. This is some characterization of the obtained nanoparticles, electron microscope images, FTIR, Raman spectroscopy. In particular, FTIR and Raman spectroscopy are confirming that we have a constant amount of the beta phase, that is the piezoelectric phase of the polymer. So our nanovectors present good piezoelectric properties despite the polymeric nature. Then we investigated the drug release, both in presence or in absence of ultrasound, so mechanical stimulation. We realized that there is a slight increment of drug released upon the ultrasound stimulation, but this is not significant. And Indeed, this was not our goal. We don't want as primary goal to modulate the drug delivery to ultrasounds, but instead, we would like to achieve cell stimulation. And indeed, cell stimulation was achieved, as demonstrated by these representative images of calcium imaging, by incubating glioma cells with piezoelectric nanoparticles, and by applying ultrasounds, we are able to activate calcium flows inside the cells. These are some confocal microscope images showing internalization in cancer cells, and particularly interesting is also Raman confocal imaging, because it's allowing, thanks to the Raman fingerprint of PVDF-TRFE, to track the nanoparticles inside the cells without any labeling. So it is a label-free tracking of the nanoparticles. And these are some experiments that are showing the efficiency of the piezoelectric stimulation in hindering cell proliferation through destabilization of the cytoskeleton and through destabilization of the activity of some voltage-sensitive channel on the surface of the cancer cells. I will not go into details of the results, but in summary, as you can see, we have a combined effect of the chemotherapy drug nuclein that is delivered by the nanoparticles and of the electric stimulation provided by ultrasounds and by the piezoelectric properties of the nanoparticles. Moving to the second class of materials that we are studying in our laboratory, I introduce a recent work about lipid magnetic nanovectors. So we are developing lipid-based nanostructured lipid carriers that are loaded again with a chemotherapy drug, again a nuclein-free A, but at the same time with the super paramagnetic iron oxide nanoparticles in order to perform both drug targeting, thanks to an external magnetic manipulation, and hyperthermia by providing an appropriate electromagnetic field stimulation. Once again, the nanoparticles are surface functionalized, in this case with the AngioPep2, in order to promote again the blood-brain barrier crossing and the dual targeting of the glioblastoma cells. This is a very quick slides showing some characterization of our nanovectors. Again, some electron microscope images, SDS page showing the efficient functionalization with the HAPO-E, and the stability, both at short-term and long-term stability of the nanovectors in relevant media. So, thanks to the super paramagnetic iron oxide nanoparticles, as I said, we are giving the smartness to our nanovector, to our nanoparticles. And in particular, we exploited the super paramagnetic iron oxide nanoparticles in order to perform hyperthermia, to locally increase the temperature, destabilize the nanoparticles, and as we will see, the lysosomes where the nanoparticles are accumulated inside the cells, induced to promote cell death. And the mechanism that we demonstrated is indeed related to the lysosomal accumulation of the nanoparticles and to the disruptions of the lysosomes upon the electromagnetic field stimulation. Here, if it works, I have a short video where we labeled the nanovectors in green, the lysosomes in red, and as you can see, as soon as we progress in the alternating magnetic field stimulation, we have a complete destabilization of the lysosomes that are discharging in the cytoplasm the enzymes, the digestive enzymes, and then contribute to the cancer cell death. And these are quantitative results that are indeed showing this double effect. So we have an effect of the chemotherapy drug that, of course, is delivered by the nanoparticles, but not only. The delivery of the drug is improved by the nanocarriers, and a strong effect of the alternating magnetic field stimulation in the presence of the magnetic nanovectors that is strongly contributing in the apoptosis and in the cell death. Now I move to the last example that I would like to introduce today. And this is a nano platform that is combining both antioxidant properties and light responsive properties, and in particular near infrared light responsiveness. Historically in our lab, we investigated since many years nanozymes, nano antioxidants, so nanoparticles that are able to behave like some enzymes that are present in our organism and that are helping to scavenge overproduced free radicals, reactive oxygen species. We investigated for many years, for example, cerium oxide nanoparticles. We performed also some experiment in space on board of the International Space Station, demonstrating the antioxidant protective properties of these nanoparticles in the muscle cells. But again, cerium oxide nanoparticles are inorganic. So if we would like to have a long-term vision of clinical translation and hopefully in exploitation for patients, inorganic nanomaterials are, of course, not the most appropriate nanovector that we can propose. So also in the case of nanozymes, of artificial, let's say, nanotechnological antioxidant, we prefer to move towards some more biocompatible, biodegradable, and fully organic nanomaterials. And this is why we proposed polydopamine nanoparticles. Polydopamine nanoparticles are very simply synthesized by starting from the monomer dopamine, and they are easily obtained in terms of these nicely distributed round-shaped nanovectors. We perform a surface functionalization with the DSPA PEG, both to confirm more stability and in case to functionalize the surface of the nanoparticles with some targeting moieties. And we demonstrated the extremely high efficiency in terms of antioxidant activities of these nanoparticles. Here, for example, you can see a quantitative evaluation performed with the EPR spectroscopy showing the strong radical scavenging efficiency of these nanoparticles. We demonstrated in vitro on a model of neuronal cells strong antioxidant protective effects, as you can see from flow cytometry data. We demonstrated that these nanoparticles can protect differentiating neuronal cells and can improve their phenotypes in terms of neurite development and length. And most importantly, we showed as they can provide also a mitochondrial protection against oxidative stress. For example, here we charged the cells and the mitochondria with a TBH, that is a strong pro-oxidant agent, and we showed that in the presence of polydopamine nanoparticles, we are able to preserve both the shape of the mitochondria and the functionality of the mitochondria in terms of mitochondrial membrane potentials. As I was mentioning, however, these nanoparticles are extremely interesting because they are also light responsive. In particular, when irradiated with the near infrared laser radiation, they are able to locally increase the temperature. So these are some images showing the local increment of temperature inside the cells, both qualitative and quantitative imaging. We used a fluorescence sensitive probe to the increment of temperature, to the heating. And this increment of temperature is indeed able not only to promote some specific function in the cells, but in the case of neurons is able to activate the functionalities of neurons by a very strong calcium flow activation. So I think I am arrived at the end of the presentation, and as I said, I prefer to give you a full vision of what we are currently working in our lab, and of course I am open for any questions of curiosity that you can have. Thank you.
Excellent, thank you so much, Gianni. So I'll just open now for questions. Anyone? Maybe I'll start, Gianni. Can you tell us about the ultrasound? What is the frequency or what is the intensity of the ultrasound which you are using to induce?
Yes, can I share again the screen? Sorry, yeah. I stopped early. So this is the protocol that we used. Maybe it was not very visible, but we are using essentially ultrasounds, and the frequency is around 1 megahertz. We tried also 500 kilohertz or something more, but actually the frequency is not so much important parameter, because of course we are far from the resonance frequency of these nanovectors. So what the nanovectors are experiencing is a wave, a static wave essentially. So it is not so much important the frequency. It is of course important the intensity, because we have to work at an intensity that alone is not able to damage the cells or to activate any other functionality of cells. We want just that the ultrasounds can activate the nanoparticles, and then the electrical cues are making effects in the cells. But we don't want any effect by the plain ultrasound. So the intensity should be carefully selected.
Excellent, thank you. All right, we have a question from Alberto. Thank you so much, Alberto, for joining. So over to you.
Thank you very much. Well, just to start, so I'm Alberto from Spain, from Gaica. So thanks for the very interesting presentation, Gianni. It's very nice and very interesting, the nanomaterials that you have developed in the institute. So my question, well, I have like more or less like 2 questions. So the first question was related with this first question as well. So about the intensity of this ultrasound, do you think, did you test, like, for example, if the intensity of this ultrasound could affect normal cells, for example, instead of just like cancer cells? And the second question is related with this first one, but like, for example, which molecules can you use to target cancer cell? Did you test, like, for example, the specificity of these particles to be internalized just like in cancer cells?
Okay, thank you very much. Yes, both questions are really relevant, interesting. So concerning the intensity, the reply is yes. We tested in normal cells, in cancer cells, and we selected an intensity of ultrasounds that is not able to induce any effect inside the cells. We also analyzed the possibility of these intensities to produce a slight increment of temperature, thanks to some fluorescence temperature sensitive dyes, but indeed we didn't see any effect at these frequencies and these intensities. So we are pretty sure that the effect that we see in combination with the nanoparticles are due to nanotransduction phenomena. And concerning the targeting, yes, of course, it is really important. Here I provided you an example where we functionalized the piezoelectric nanoparticles with the ApoE, that is a short peptide that is targeting the endothelial cells on the blood-brain barrier, but at the same time is also performing a dual targeting task, because the receptor is overexpressed also in cancer cells. And in the past, we also performed some trials, and we published a study concerning the functionalization with antibodies. and more specifically with the antibody against the receptor of the transferrin, and it worked pretty well in vitro. We decided to move towards short peptide and short ligands because we think that moving in vivo, this can have an improved specificity and also more stability.
Excellent. Thank you so much, Gianni. Is there any other question? Yes, Marcus, please.
Hi, thank you very much for your interesting talk. I saw in one of the earlier slides that there was a particle system that delivered therapeutic peptides, if I remember correctly.
Therapeutic peptide, actually no. The peptides are used as a targeting agent.
Right.
So we surface functionalize with these short peptide that are able to target the nanoparticles towards a precise class of cells. But the therapeutic agent in both the example that I showed you is a neutrin 3A, that is a non-genotoxic chemotherapy agent.
Mm-hmm. And does your group or your institute do any research using peptides or therapeutic peptides?
I think absolutely yes. Maybe even PAOLO can tell you something more about this, but in institute we have, for example, a group that is focused on therapeutic polymer. So I think that absolutely they are developing something. Maybe we will not see today, but there are some groups that are working in these directions.
Okay, thank you. I might have just one more. I wanted to understand how you make your polydopamine particles. What's sort of the synthetic procedure to get them?
It's a very, very simple procedure based on a Sturber reaction. So we solved the monomer, the dopamine, and then we had ammonia at different ratio in order to achieve a different size of nanoparticles. The reaction is performed overnight, but it's really, really simple. It is based on a Sturber reaction, like to synthesize silica nanoparticles or something like this.
Okay, thank you.
You're welcome.
Excellent. Thank you so much. Any other question? If not, thank you so much, Gianni, again. So now we will move to my presentation.
All right, whiteboard, multiple things. I guess I need to go here. And then I need to move here, sorry. All right, let me close this. We're getting there. Can you see the full-size screen or just a portion of it?
Not yet full size, but it's coming.
All right. Okay.
Yes, now.
There we go.
I will interrupt you 5 minutes before the end of the 20.
Thank you. Thank you. And I know that you are quite precise, accurate, and also brutal in the interruption. So I will try to make sure that I can actually, I will try to stay in the time. So the laboratory that I originated in about 6 years ago, 2015, in Genova, the Italian Institute of Technology, and that is called Laboratory on Nanotechnology for Precision Medicine, is actually working on the development of different type of drug delivery systems. So we don't focus only on one single configuration, but we like to basically span, you know, the different dimensions across this virtual yellow ruler that you basically see in front of you. And so we go from conventional micelles and lipid and polymeric nanoparticles, which are, you know, again in the 100 to 100 nanometers in size, and they are relatively standard system, again based on polymers and lipids. Some of them have actually hybrid systems with the lipids and polymers combined together. But we are also working with systems that are definitely larger than that. They are in the micron size. These are the systems that we call discoidal polymeric nanoconstructs. They are made out of polymers and they are not spherical in shape anymore. We then work with the larger, even larger system, microplates and micromeshes, which are no more injectable. So all the way up to the DPN, so about 1 micron, 2 microns, whatever is basically on the left side and side of the ruler can be injected systemically and navigate through the bloodstream. Whatever is on the right-hand side, on the other end, the microplates and the micromeshes are basically implantable system. Here you see some images taken at the electron microscope of the 3 different classes of particles, DPNs, microplates, and micromesh, which are a little bit less conventional as compared again to the spherical nanoparticles. And also at the bottom, you have a synthesis somehow, a summary of how the groups operate. So the group is highly interdisciplinary. We have people with a background in engineering, with a background in chemical physics, and many people are actually, some of them are actually working on computational modeling, and we do a lot of computational modeling at different scales. We go from molecular dynamics all the way up to continuum mechanics, depending on the problem that we need to solve. We develop also our own microfluidic system in addition to the standard in vitro assays that can be done in order to characterize the particle functions. And then, of course, we do also our in vivo experiments. So basically, we have, again, engineer, physical chemist, veterinarians, biotechnologists, and molecular biologists that are working together in the same group to address the different issues that we want to solve. Today, in the interest of time, I'll be focusing only on 2 major platforms that we have more recently developed in the lab, and these are the micromesh and the microplates. The micromesh has been mostly developed recently for the treatment of brain tumors as an implantable system. The microplates, I'll give you one of the an example of one of the diseases on which we are focusing, which is osteoarthritis. So it's an inflammatory chronic disease that is limited to the synovial cavity and the knee in the joints. So the first example is, as I said, related to the micromesh. I don't have to go through the details of why it is important to work on brain cancer and in particular glioblastoma. You can actually have a look at these curves here that are showing the incidence and the mortality associated with brain tumors and with glioblastoma over the last 20, 30, 40 years. And the curves are pretty much flat, meaning that nothing has changed over these numerous decades, neither in improvement in early diagnosis and so prevention of the disease, nor in treatment of the disease per se. And this is one of the reasons why we have been focusing over the last years on glioblastoma. I would say 10 years over the treatment and trying to find therapies for glioblastoma. There are very limited therapies that are actually functioning. The STUP protocol up here is basically the protocol that is used in the clinic for the treatment of glioblastoma and other brain tumor. And it's basically surgery followed by adjuvant radiotherapy and chemotherapy with temozolomide, which is a drug that has been approved by FDA at the end of the 1990s. So we are basically using a protocol that is 15 years old and more with drugs that are a couple of decades old. Gliadel wafer, which is basically the only approved wafer that is around and out there, has been again approved by FDA at the end of the 1996 and has not provided any significant improvement in therapy. The other interesting thing is that more recently, big trials in clinical trial 2 and 3 level, phase 2 and phase 3 have been failing with immunotherapies, which seems to be the great new therapy that is outside there for defeating cancer. And simply because large objects like antibodies and large molecules cannot really cross the blood-brain barrier, or at least that it cannot cross a mild mild permeable blood-brain barrier. And even if they can cross, they cannot permeate deep into the tissue and then basically attack most of the tumor. So this is the reason why over the last 4 years, this is a work that actually took about 4 years, we developed the micromesh. And these are two cartoons that somehow represent on one side the architecture geometry of the micromesh, and on the other side the application of the micromesh. Here you see a cartoon showing the structure of the micromesh. The micromesh is a compartmentalized system that comprises a polymeric mesh made out of PLGA, which is the green network of strands that you see here, very tiny, 3 by 5 microns, that can be loaded with hydrophobic molecules. In the current configuration is loaded with diclofenac, an anti-inflammatory molecule, but can be loaded with curcumin and other, hydrophobic molecules, including docetaxel and so on. And this mesh is actually deposited over a very tiny microlayer that you see here in azure, light azure, and is basically made out of PVA, which being a very hydrosoluble polymer. And within the PVA, you can actually disperse another therapeutic agent that is typically hydrophilic, and in the current configuration, we have dispersed nanoparticles loaded with docetaxel. So you have diclofenac that is dispersed in the green mesh, and the nanoparticle loaded with docetaxel that are dispersed in the microlayer. So you have a two compartmentalized system with two different drugs that are loaded independently and can be released independently following different release profiles. The micromesh is applied over the surface of the resected cavity of a tumor, in the case of glioblastoma, pretty much like Gliadel, but has some specific features that I will show you in the next few slides. Here you see another representation, and in particular, you can actually also have a more clear more details about the actual geometry in terms of size. You have this 3 by 5 micron, as I was telling you, the strands, and then the openings are 20 microns. Very important size because it's somehow comparable with the size of a couple of tumor cells. And this is very important for the way the micromesh works. This is confocal microscopy image. The mesh is in green because loaded with curcumin. You can see in the background red dots, which are associated with the nanoparticles. And then you have an EM image that actually shows, again, the walls of the micromesh of the PLGA strands, and then the nanoparticles that are actually coming off the PVA as it dissolves in water. Additional representations and images, EM and confocal microscopy that actually show the PLGA mesh per se. You see how tiny it is, and the PVA layer. Within this mesh, we have the diclofenac. Within this micro layer, we have the nanoparticles. The fabrication process is relatively straightforward. It's based on basically replica molding, starting from a silicone template, becomes a PDMS template, replicated in a PVA template, and then on top of this PVA template, we deposit a PLGA paste that is enriched with the molecules that we want to deliver. So relatively straightforward, working already on big wafers of 8 inches, and so it's actually a macroscopic system that can be realized in a relatively short period of time. One of the features of the micromesh, as I said, is the ability to have this PLGA mesh that can actually be released progressively as the PVA layer is dissolved. And as that happens, what happens is that whatever is loaded in the PVA is releasing the surrounding environment. So basically on the tumor bed, basically. And the mesh is also free now to basically deform and conform to a complex surface of the resected cavity of the tumor. And so what you see here in these EM pictures and then in a confocal microscopy image on the right-hand side is actually a mesh that is wrapping around the tumor spheroid. It's literally wrapping around and intercalating with the tumor cells. This is very important because the mesh is carrying the drug that is then supposed to kill the tumor cells. So the mesh is actually wrapping around the tumor spheroid as a fish net would do around a school of fish. And so again, this is one of the unique features of this system, and is extremely important because this intimate interaction dictates also the therapeutic efficacy of the mesh. And when you compare the ability of the mesh to contain the growth of a tumor spheroid, and you compare the mesh with a flat PLGA layer, so no holes, just PLGA, very thin still, but not capable to deform. And if you realize that the PLGA is so-called flat in a way that it releases the same amount of docetaxel with the same release profile, so that you can do a nice comparison, you basically see that the micromesh outperform the PLGA flat easily. Another advantage of the micromesh is that the nanoparticles that are released by the PVA micro layer, once they're released in solution, they still have a very tiny nanoscopic PVA layer on the surface, as it has been demonstrated through a careful chemical analysis of the surface of the particles before and after being released by the PVA microlayer. And this is extremely important because we believe that the presence of this tiny PVA layer on the surface of the particle can facilitate their permeation deep into the tissue, like the super lubrication that usually is associated with the pegylation of the particle. And in fact, when we actually compare the permeation of these particles into a tumor spheroid, on the right-hand side here, you see basically spheroids that have been exposed to particles that were released from the PVA layer versus particles that were not released, the same one, but not released from the PVA layer. And you basically see that there is a significant variation in coloration of the spheroid. In this case, you see dots going all the way down. So red dots, which are the nanoparticles, going all the way down into the tumor spheroid. Here, basically, everything is pretty much dark beside the surface of the spheroid. When you do this experiment in vivo, in mice, using intravital microscopy, you can actually monitor over time the permeation of the nanoparticles into the tissue, and you clearly see over time how the gradient is actually reducing, of course, because it means that the particles are distributing down into the tissue. But at the same time, even at 10 days, you still see a significant gradient in concentration that tells you that the particles are still moving and still permeating in it over time. When it comes to in vivo, we did multiple and different type of experiments using, of course, orthotopic glioblastoma models where the tumor cells are inoculated directly in the cranium. And we used two different models. One where we don't remove the primary mass, and then one in which we actually remove the tumor mass, which is actually what a neurosurgeon will do before the patient goes through adjuvant radiotherapy and chemotherapy. We left the cells grow into the cranium for about 15 days to make sure that the tumor, reach a stable size and can actually grow. If you do it for a shorter period of time, more often you have a resolution of the disease in the sense that the tumor is not really stably growing. And then we consider two different types of cells. We consider U87 and G cells, which are a classical cell line for glioblastoma, and also human cancer stem cells that were given to us by patients, by hospitals that were treating patients affected by glioblastoma. And what you notice in this plot is that here you have a huge number of control experiments, but you notice that the micromesh is basically outperforming all the other control groups. Perhaps only the docetaxel micromesh case is actually doing quite well. When you remove the tumor mass, first of all, notice that the animals are still dying, which means that the tumor margins were still there, were growing, and were then taking the animal to death, just like it happens with the patients. Unfortunately, you see that TMZ, temozolomide, the classical gold standard, is doing better, of course, of the control. Then nanoparticles, low-weight docetaxel, injected directly into the cranium through the orifice of the tumor, are doing better than TMZ as expected, but the microplates is doing it way better. And consider that here we have the same nanoparticles of these yellowish line, but they are not injected with a syringe intracranially. They are released over time, and they are permeating more uniformly and more efficiently the tumor. Same trend when you actually consider the human cancer stem cells model. The time is, it just takes longer because these human primary cells takes a longer time to grow and develop into the cranium of the patient. Another thing that you might want to notice is the duration of these experiments. We have been following animals for almost 270 days, 300 days. So we're looking at 9 months of evolution of the disease with the observation of the animals and no sign of major complications. The second portion of the story is related to what we call the microplates and their application in osteoarthritis. I don't have to give details about the disease. You know that it's an inflammatory disease, mostly inflammatory process that actually develops into the joints and progressively leads to degradation of the cartilage, death of the chondrocytes, inflammation in the synovium, and so on. So here we are using a different type of system, and these are called the microplates, which are no more a continuous mesh, but they are discrete microparticles. And you will notice, you might notice that this is basically the negative or positive sides of the micromesh. So the micromesh is actually taken from this template here. We do an additional step and we basically release the microplates. So the micromesh and the microplates are basically two different phases, if you want, of the same coin. So these microplates are made out of PLGA. We're doing also them with other polymers, and using different we're always available to use different polymers. So I know that there are experts in polymer chemistry and polymer synthesis. I will be more than happy to discuss also the use of different polymers. But basically the idea here is, again, to use a template that we fill with the PLGA paste. We're doing hyaluronic acid, chitosan, mixture, alginate, gelatin, and so on. It can be done with many different systems. You name it, and usually it's relatively straightforward to change the polymer. And you end up having these particles that can actually be tuned in height and also in geometry. These particles, as I said, can come in different geometrical, with different geometries, with different amount of PLGA that will change actually the biopharmaceutical properties as well as the mechanical properties. And the mechanical properties, when it comes to joints, is very important. Here you see that we can actually modulate the Young's modulus of the particle. We can go from very soft to very rigid. And it's very important, of course, because these particles can actually also act as a cushion within the synovial cavity. You can load them, in this case, with the dexamethasone, a classical conventional drug that is used for the treatment of osteoarthritis. You can release them and check how they are released more rapidly, less rapidly, depending on the environmental conditions. You can test the efficacy of dexamethasone microplates on ATDC5 cells, which are chondrocytic cells, and show that upon stimulation with LPS, you can actually alleviate the inflammatory process by treating them with DEX microplates. And then, of course, the following step is to inject them intra-articularly into the joint and try to understand what happens in terms of response of the animal to the treatment. And this can be done by looking histopathologically at the, slides with the eye of a professional histopathologist and pathologist. And there are different scores that can actually be provided in order to quantify the effect of the treatment. One of these is the DJD score. And here you can see that basically an healthy animal return a DJD score of close to 1. And when you treat the same animals with microplates, one single injection after 30 days, this is one single injection after 30 days, you basically see that there is no big difference between the healthy animal, the DEX microplates, and actually also the empty microplates are having some effect, and we're still investigating the cushion effect that I was telling you about. Other scores like the ORC scores are again showing that the DEX microplates are actually working quite nicely in terms of containing the disease and preventing the degradation of the cartilage, and so ameliorating the conditions within the joint. I think that with this, I can actually close, presented 2 different platforms. This is a picture of the group well before the COVID pandemic. I guess it was back in 2019. Some of the people that left, new people came in, and also a display of all the fundings that have supported us at the European level and at the Italian level. Thank you.
Thank you very much, Paolo. I think we have time for a couple of quick questions. Marcus, please.
Thank you. Thank you for this interesting talk. I wanted to maybe go a bit deeper into the those cushions that you were describing at the end. I have 2 questions. The first one is a quite straightforward one. How small can you make these platelets?
Yeah. So the microplates are done through a template strategy, and so this means that we can go down to several hundreds of nanometers. So let's say 500, 600 nanometers. There are other techniques to go lower in size. I mean, the print technology, for instance, from Joe DeSimone goes down to 50 nanometers. So. But our objective was not that of going down in size. We wanted to actually be into the micron size. 20 microns, these microplates are 20 microns by 20 microns by 10, 20 microns. So they are comparable in size to the size of a cell. And inside them, they can actually store a large number of molecules that can then be released over a long period of time. I highlighted that the results that you saw in vivo were after 1 month and 1 single injection. So the dexamethasone was actually sustained and continuously released for 30 days out of those microplates into the synovial cavity. So if you start using particles that are smaller, the release profile will be much faster, and everything will be basically you will release all your drug in a few days. There is also another important thing that you need to consider when you work with osteoarthritis is that there is a lot of hyperpermeability in the walls of the synovium, and because of the inflammation process. And so even particles of 100, 200 nanometers, they don't stay longer. The dwelling time within the synovium is actually very limited also for larger particles. So for this reason, we have been focusing on the 20 microns size. And right now we are characterizing all the mechanical properties of the system using more conventional tribometric approaches and tribological approaches to try to elucidate the effect of the empty microplates. So that cushion effect.
Yeah. Thank you. I have another question, but maybe let's first go to Anna, and if there's time, I'll ask that one.
Are you sure?
Okay, please,
Thanks.
Anna.
Yeah,
Thanks, Marcus. So yeah, fantastic talk. It was really interesting. I was
Thank you.
Wanting to ask about the sort of tuning the properties of your meshes with the different combinations of the degradable polymers, and does that change the sort of properties of it, and would that be a way to apply it to different applications in terms of the mesh degradation and the mechanical properties of the mesh?
Yeah, well, first of all, beautiful background. Is that a thrombus?
Yes, it is, and you'll see from my talk later. This is
all right.
This is what I do.
Fantastic. Yeah, because we are also very much interested in thrombosis and stroke.
Yes.
Anyway, right now we have been working for the micromesh. We have been working mostly, actually, only with PLGA and PVA. The mechanical properties of the system are very much related to the level of, the thickness of the PVA and the molecular weight of the PVA, the amount of PVA that you actually, the density of the PVA, right? The concentration of the PVA, as well as the thickness of the PLGA mesh. So with this system, if you want to actually modulate the stiffness and the flexibility, the only way to do it right now is by changing the geometrical properties. And this is something that we are actually investigating. However, I agree with you. If we move away from PLGA and we start using other type of polymers, there might be, a broader range of flexibilities and mechanical properties that can be explored. We haven't started that yet, but we're going back to the microplates. We're making microplates with hyaluronic acid. We're making microplates with chitosan, alginate, and the same technology can be translated to the micromesh. But we're very much interested in exploring different type of polymers and for realizing these systems.
Fantastic. Yeah, that sounds amazing.
Thank you very much. I think that we can move to the next speaker because our delays is increasing, in particular for the Australian friends that are quite late in the afternoon otherwise. So please, now is we are moving from a very, very long title to a very short title. So please, Marcus, with your talk, Molecular Polymer Brushes. Thank you, Marcus.
Thank you very much for the introduction. So I noticed in the talks before there were quite a lot of very specific applications, a lot of progress towards them as well. So I'm not sure whether I can follow in those footsteps, but I'm going to be talking a little bit about a platform or a workhorse that we have in my group, which is the molecular polymer brushes. But before before I go into this, just a very brief overview of my group. So I'm in the School of Chemistry. I'm a polymer scientist, and the group is generally built on four pillars. I guess the ones that are most relevant for today would be polymer self-assembly and polymers in biological studies, rather broad terms, but that's sort of the things that I would be focused on today. But we've been quite active as well in the synthesis of polymers, particularly using photocatalysis or heterogeneous photocatalysts to drive radical polymerization, as well as focusing a bit on hard matter or finding ways to structure hybrid and inorganic materials, particularly for battery electrodes and also water splitting. But today I shall talk a bit about molecular polymer brushes. So for those who haven't come across these, you will be all familiar with polymers or with macromolecules. If you have a macromolecule, put it in solution, it will coil. You get usually sort of approximated sphere, maybe around 5 nanometers or smaller. But what you can do now synthetically is you can attach side chains to this coil. And if you manage to do this in a very high density, so you have a chain crowding effect, you actually force this backbone to stretch, and then you end up with what's called a molecular brush. It is, however, still one macromolecule. So all the chemistry that you can do to small molecules and linear macromolecules, you can do also to a molecular brush. But what changes now is really that you are no longer a 5 nanometer object. You are now actually something that is 20, 30, 100, 500 nanometer object. So you're becoming a nanoparticle despite being one molecule. And of course, this lends itself then to the use in nanotechnology or nanomedicine. And I'll show you also this AFM here because just to demonstrate the uniformity by which you can make this material. So each of these white worms is basically one of these macromolecules. In this case, they are in cylinder shape. So you can see each molecule individually with AFM. I spare you how we make them. So there's a lot of quite, I guess, nowadays established grafting methods that we can use to build up these these molecular brushes. But what we can, and we can rely on these nowadays. So it's really I guess the approach of this subject is now more sort of what particle do I need to build for a particular application. And you can see here various compositions. The compositions can be achieved, block type, core shell, etc. And these are all actually unimolecular objects. So they're all covalent. So they don't experience sort of dilution effects or fall apart. So obviously in the context of drug delivery or nanomedicine, this is a very interesting particle. But also you can use them as building blocks. So these are they behave the same way as linear polymers or block copolymers, but they have much, much higher molecular weights. So if you think of a self-assembled construct, it has much higher stability. So for example, a micelle made from molecular brushes would have an aggregation concentration of maybe 543 nanomolar. So if you dilute this by injection into a bloodstream, for example, it will not fall apart. So the reason why we work with these materials is because we think these are very customizable polymer particles. So not only can you make them rather uniform, but it offers a very high degree of control. And you can control various design parameters such as size, shape, aspect ratio, composition, surface, chemistry, also stiffness. And often what you can actually do, you can do this independent from one another. And our workers is the grafting from approach, where you basically use a polymer backbone and then you grow side chains from it. And via this approach, you can introduce a lot of different functionalities for radio labeling, for fluorescent labeling, but also for drug attachment if you choose so. And this is just another AFM to show you how easy it is for us to just change essentially the aspect ratio. So going from a sphere-like object on the left to a rod-like and then to a filamentous object on the very right. This slide is here more as a disclaimer. So while we are motivated sort of to push in the area of nanomedicine, we are not really a group that does specialize in drug delivery. There are many people who know this much better, but we have done a proof of concept study where we have attached cytotoxic drugs via a pH cleavable moiety and then followed the pH or the pH triggered release intracellularly with fluorescence lifetime imaging. So it is possible to use our system for that, but we have thus far not explored this in detail, but of course are very open to work with people in the drug delivery space. And the reason why we are not necessarily delivering drugs right now is because we don't want to just add another nanoparticle to the vast array of nanoparticles that already exist. So if you look at the evolution of polymer nanoparticles in nanomedicine and also the ways by which we can control nanobio interactions, you'll see that there's a vast amount of literature. And of course, a lot of it is actually based on micelles or vesicles or dendrimers. And this is because these materials are rather simple to make. They're also often spherical. And what we have learned here is, to a decade worth of comparison studies in a way, is what size is important, for example, in an in vivo setting, also what surface composition is important for successful drug delivery or for targeting. But if you look at the current area in polymer nanomedicine, what has emerged is really that other factors or other design parameters such as shape, aspect ratio, stiffness are equal determinants of the outcome of your in vivo experiment, for example, as is size and surface composition. So this makes the whole picture a bit more complex. And if we were to continue like we've done in the last 30, 40 years, doing comparison studies with various particle systems, then I think it would take a very, very long time to understand this complex picture. So having one platform where you can alter design parameters independently from another, I think brings in merit and benefit. And particularly if you're going to look even further ahead when you're looking at personalized medicine or mimicking nature in a way. So for those who are a bit familiar with polymer chemistry, you can see here that our particles, they are mainly made from PEG-based materials or PEGylated materials. We can, of course, switch this to be polyoxazolines or zwitterions, so the biocompatible component of the brushes can be easily changed if we wanted to. And also what we usually do is we add a bit of functionality for radio labeling or fluorescence labeling in our case. So this slide is essentially here to demonstrate the simplicity of how we make our materials. So basically starting of a polymer chain with a repeat unit of X, we then, as I said before, graft our side chains from it to establish our brush. And simply by varying this X here, so the length of our backbone, we achieve different shapes or different aspect ratios. So basically you have a sphere or rod or filamentous objects, but they're all now identical in composition, they're all identical in diameter, and so and also in terms of functionalization levels. So it makes them very comparable if you were to investigate their use in a cell culture or an in vivo setting. And sort of the first contribution we had here in this space was a few years back when I originally came to Australia. I joined a very bio-focused group, and so I had already experience working on these brush systems, but we sort of reconfigured that maybe there's some merit in using them also in an in vivo setting. There were no studies back then on this, so we established essentially a pharmacokinetic profile and biodistribution of these materials, and particularly first looking at the various shapes. It was not too surprising, I guess, that the bigger or the larger ones, the filamentous ones, get cleared a bit faster than the shortest or sphere-like objects. But in all cases, we actually saw that they have quite competitive half-lives, up to 31 hours for the smallest component. And they ended up in RAS system, so liver and spleen, as to be expected, I guess, for nanoparticles. So with having long enough circulation time, we then moved on to have a look at tumor-bearing models. We saw similar trends as we saw in the red study that I showed on top in terms of bioavailability of our materials. But also what we saw, again, aside from getting into a liver and spleen, that we did actually manage to get into tumor tissue. And we did a comparison study with dendrimers and also with pegylated nanoparticles, and we used them in the same sort of tumor-bearing models, and we saw that we have quite a comparable outcome in the end. What we also see here within errors, there's no real difference actually in terms of amount of materials depending on the length that actually ends up in the tumor. So we wanted to have a closer look at the tumor tissue then, and to understand where our particles actually are going. So particularly look at extravasation, so they're moving away from the particles, from the blood vessels into the tissue. And we had sort of between 5 to 7 micrometer on average, the distance that particles managed to move into the tissue. But again, we didn't really see too much of a difference actually between our different shapes. Which then meant we wanted to look at this a little bit closer in an in vitro setting, and to maybe study this a bit more thorough. So we moved to looking at multicellular cancer spheroids, some we saw already in earlier presentations. And just as a first test, of course, we incubated various concentrations of our materials. So in this case, the rod-shaped particle. You can see, of course, with increasing concentration, you get much more association and higher fluorescence, which is not too surprising because you're supplying the system with more brushed nanoparticles. The interesting part was really when we compared again our shapes, that the filament and the sphere had vastly less association with our tumor spheroid. And this is particularly interesting because when you look at the brush particle number, because we're injecting by mass, there's actually so much more sphere nanoparticle around in the system compared to the rod one. And this also translated into tissue penetration, where the rod penetrated further into the tumor spheroid compared to the other two shapes. And nowadays, there's actually a lot of studies in literature, particularly on polymer nanorods, corroborating essentially what we see here with our brush system as well. But also we saw what I mentioned on the slide before, that this does not necessarily translate in our in vivo setting. The other thing that we recently looked into is stiffness effects. So if we have, as I mentioned to you earlier on, the brush system allows us to vary particular design component almost independently from another. So we can make nanorods that are identical in volume, in diameter, in length, aspect ratio, surface composition, but they are slightly different in terms of their mechanical property or their rigidity. And we achieved this by introducing a small high TG core. And we also assessed actually with small angle Newton scattering to put sort of a numerical value to their differences, so a particular focus on persistent length. And what we saw when we incubated these now with 2 different cell lines, so colon cancer and lung cancer cell line, is that the soft nanoparticles and the rigid nanoparticles behaved very differently. You might not fully see this now on this overview slide, but if you zoom into 2 of those, you'll see that the soft one shows more sort of spot fluorescence once they get internalized, whereas the more rigid one show actually more sort of tubular fluorescence. And if we then do a mitotracker assay, we actually realize that our rigid particles exclusively interact with mitochondria. And the reason why I find this interesting is because it's supposedly a mechanical cue that actually dictates the intracellular fate, because in terms of chemistry and also the overall dimensions, these particles are identical. And the other thing that we're currently looking at is amphiphilicity. So with self-assembly, this is not very easily studied because if you can imagine, if you change the amphiphilicity of your, say, polymer, it will affect the outcome of your self-assembly. But with our covalent construct, we can basically start off from a very hydrophilic object or nanoparticle and then progressively ramp up the hydrophobicity. This is shown here basically by D0, which means no hydrophobic component, purely hydrophilic nanoparticle, and then we add various percentages of hydrophobicity to it. What we can see in the 2D monolayer experiments, so these again colon cancer cells, if you get to a certain level of hydrophobicity, the particles do not want to interact anymore with cells, so it cannot be internalized. We also see in some way that there seems to be a further or an increase in association when you ramp up the hydrophobic component. We also see effects of these in the 3D model, so where again we see the most hydrophobic nanoparticle doesn't actually interact anymore or penetrate into spheroids, whereas hydrophilic ones or progressively more hydrophobic ones still manage to interact, associate, and also penetrate into our spheroids. We took this one step further actually into in vivo, and this is unpublished data, but it's hopefully close to be finished. And so as you remember from the very first in vivo experiments, I guess we've done it many years ago, we saw our particles go into liver and spleen by default as they are nanoparticles. But when we look at this from the increasing hydrophobicity context, then we can see actually once they get to a certain level of hydrophobicity, they almost exclusively go into the spleen. You could almost speak of spleen targeting in this case. You can also see that
5 minutes, Marcus.
Yeah, thank you. You can also see that with increasing hydrophobicity, you are ending up much more in the liver. And that might not be too surprising, since these are the organs of responsible for clearing. If we now go to the tumor, however, it's sort of the opposite. You want to have a hydrophilic particle that actually allows you to go into the tumor. And these are now data after 24 hours. Whereas if you have it progressively more hydrophobic, you're actually getting less into the tumor until a point where almost not as much goes in there anymore, which might be also due to the particles being cleared from the body. But the really interesting thing is, I don't show this now here on the slide, but when we follow this over a few days, what you can actually see is that the hydrophilic particle moves out of the tumor again, it gets cleared, or at least the content goes lower, whereas the D20 and the D40, so the particles that have some level of hydrophobicity, actually increase the percentage that that is located in the tumor. So we're thinking in a way that by carefully playing with the amphiphilic balance, you might actually improve tumor retention or tumor homing. So that's sort of a summary on that part here. So we use our molecular brush platform in a way to fundamentally study structure function property relationships of these types of materials. And we've established that we have competitive circulation time. We can extravasate and accumulate in tumors. We have seen that shape can have an effect. However, there is sometimes a discrepancy between in vitro and in vivo experiments. And we also showed very recently that small additions of hydrophobicity may actually improve tumor homing or tumor retention. So the last piece missing is now looking at grafting density and topography, which we're doing at the moment, with the ultimate goal to essentially pick the design parameters that worked best and combine them into one particle. So to look for synergistic effects, and then of course down the track to use them also in a drug delivery context. And in the last 2 slides, I just want to sort of highlight a few things where there might be opportunities for maybe collaboration. So I mentioned just then that we're looking at nanoparticles with the topography, and in soft matter, it's not very straightforward to make nanoparticles that have a surface topography. This is sort of somehow how our particles look like. So we can make cylindrical objects that have protrusions. You could almost think of like viruses sometimes have. And we're very keen to explore the applications of these materials further, maybe even looking at artificial virus particles. So in general, I guess the invitation would be for anyone who is interested in using our brush system or our self-assembled brush systems in a drug delivery context. Particularly a reminder that the results that I showed you with targeting mitochondria, or in a way spleen with the hydrophobic particles, or of course going into tumors, that would be interesting to us. The other thing that would be interesting to look at flow. And I know Anna Waterhouse is obviously working also on flow. So trying to understand a little bit more how the shape affects the flow, and maybe even looking at margination, when at what size the margination will switch on or can be exploited. And then a little bit more sort of blue sky, or things that I don't currently do in my lab, but I think we could use the expertise from my lab to work further. And this is why I asked in the first talk about peptides. There's mounting evidence that if you have therapeutic peptides built into an architecture, you're switching on new properties. First of all, you can now do endocytosis, which you often can't do with a small therapeutic peptide, but also you have much higher hydrolytic stability. And I think borrowing some of our concepts, we could easily make brush materials or architectures of peptides and use them then for delivery application, and also borrowing some of our photocatalysis work where we can make polymers in a very mild, visible light driven context, and also in water. That would be our Angewandte paper from 2 years ago. And the other thing that would be interesting is what I say here, multivalency approach. So as you can see with brushes, you have many side chains, and they're actually all accessible at the terminus via click chemistry. So we could design materials that have multiple copies of a particular peptide or motif. Of course, for targeting, this is very interesting, but also the example that I show here is that there are peptides, such as from the Scripps Institute, where you can, for example, induce cholesterol sequestration. So could one make synthetic materials that in a way act as artificial cholesterol efflux agents. So exploiting the multivalency approach a bit further. And with this, I'll go to the acknowledgement slide. So thank you, obviously, to my group, in particular the students Theo, Faraz, and Antoine, and Sarah, who have done the work that I presented today, and the various collaborators and people who supported us along the way. And thanks very much for the opportunity to present to you.
Thank you very much, Marcus. Really interesting talk. Are there any questions from the audience?
I would have one, actually.
Yes, please, Teresa.
Thank you. Marcus,
Yeah.
do you have, first of all, thank you very much. Very interesting stuff. Do you think it is possible to co-assemble within your polymer or outside your polymer nanoparticles, inorganic nanoparticles? Do you have experience on that?
That's totally feasible. Things that I haven't obviously presented today is you can use these brushes also as nanoreactors or templates. So it's very straightforward, for example, to make a core shell corona object where you selectively fill a particular compartment to realize, for example, nanotubes down the track. This can be done either by small molecules that you incorporate, or even by nanoparticles that you then tell to go into a particular compartment. And through that, you can then arrange nanoparticles in 1D, in 2D, or in a sphere essentially. So that's totally possible.
Thank you. Are there any other questions?
Okay, will be very interesting.
Sorry, Teresa, I think you lagged at the end, so that's why I couldn't really gauge whether the answer hit the spot or not.
It's not just. Oh, sorry.
Yes,
just a quick question from my side. You were mentioning also mitochondrial targeting. Did you really perform some testing? Because it is quite interesting for us.
Yeah, we did essentially the colocalization, various colocalization assays, and then look at the overall overlap of the fluorescence from our brushes and also from the mitochondria, and then mitochondria and brushes, and these did confirm that these do go into mitochondria or are colocalized around the mitochondria and not.
And sorry, which targeting ligand did you use?
So that's the thing. We have no targeting ligand. So I know there are a few studies recently been published that sometimes Cy5, so red dyes that carry a positive charge, can actually lead to mitochondrial targeting. But we have the same dye on both objects. The only difference of the nanoparticle is the rigid core.
I see.
And so that's what I mean by it's in a way, it's a mechanical cue that tells the object to go to the mitochondria, which is, we only published this early in the year. So that's still sort of a question mark that we would like to explore a bit further, because we don't understand why this is happening.
Really interesting, really. So thank you. I think we are at the end of this first section. We are slightly delayed, but I think we can make a 15 minutes break. So here in Italy is 10:10. We can meet again at 10:25 here in Italy. That is 6:25 p.m. in Australia, if I am correct. Okay.
Yes. No problem. All right. So see you in 15 minutes. Thank you so much.
15 minutes. Yes. Thank you.
Gianni, facciamo una prova di sharing o ci fidiamo?
Ah, ciao Guglielmo, buongiorno.
Ciao. Sì, mi vedi qui solo perché poi starò in piedi, no?
Sì, vedo a malapena la fronte.
Sì, lo so, no, sono qui. Ah no, ecco, adesso posso fare lo sharing, quindi vediamo un po'. Così tu vedi l'orso, giusto?
Allora, sta arrivando qualcosa, vedo l'orso col salmone.
Esattamente, ha preso il salmone che c'era la marea. E così vedi la presentazione?
Sì, perfetto.
Ok, allora non so, la lascio lì o la tolgo?
Ma magari toglila, non so se Wojtek vuole fare visto che farà lui il chair, un 30 secondi di presentazione. Tanto dovrebbe essere abbastanza rapido. Grazie di nuovo per essere qui.
Ma no, figurati, scusami, anzi che sono arrivato in corsa, ho fatto un'ora invece che 2 di lezione, perché poi quando tu me l'hai chiesto non sapevo l'orario, quindi poi non ho scelta. E fra l'altro poi temo che dovrò scappare, cioè non mi piace fare l'uomo così di spot che appare e scompare, però purtroppo...
vabbè, è così, dai. Non c'è problema.
Perché questo è un altro degli aspetti negativi di fare online, che se io fossi in Australia starei lì e non avrei altro da fare. Invece lì, già ho messo un cartello alla porta, perché sennò...
Dai, speriamo di ritornarci presto all'epoca pre-Covid.
Sì. No, dovremmo, dal 15 ottobre, secondo me anche noi stiamo...
Sì, io dovrei essere in Giappone dal 15 ottobre.
Ah però, bravo.
Per 2 mesi e qualche giorno. Vedremo. Sto aspettando il visto, perché con il fatto del Covid è un po' problematico spostarsi.
Immagino. Io vorrei andare a Boston, se riesco.
Ma ho sentito, io non so che vaccino tu abbia fatto, che non riconoscono AstraZeneca per il momento.
Sì, è vero. Infatti io sono AstraZeneca come la maggior parte degli...
Anch'io.
Sì, però a me hanno detto che in realtà... Dunque, il consolato dice che faranno entrare tutti i vaccinati, quindi. E Fauci ha rilasciato un'intervista in cui ha detto la stessa cosa. E poi io pensavo che loro lasciano fuori l'alleato inglese, non mi sembra credibile.
No, infatti, speriamo, perché noi dovremmo ritornarci il prossimo anno, starci un po' di nuovo a Cape Canaveral, però se non aprono ai vaccinati, perché noi siamo tutti AstraZeneca nel mio lab, perché
Sì.
siamo stati fra i primi appena possibile a farlo, e AstraZeneca era ancora legale agli under 60. Quindi...
sì. No, infatti. Tu, fra l'altro, adesso hai abbandonato un po' il territorio?
Sì, ma ormai da 2 anni.
Non fai più niente con loro?
No.
Ti hanno disgustato? No,
ma no, è stato un po', sono tutta una serie di motivi sono stati. Un minimo disgusto c'era, però ecco, non è stata la decisione preponderante. Diciamo che la decisione principale è stata...
Sei un IT puro.
Sì, sono IT puro. C'è stata una decisione più che altro personale, che gestivo male il pendolarismo, iniziava a diventare stancante. Poi, vabbè, mia moglie è venuta in Italia finalmente, perché lei è straniera, abbiamo preso casa a Pisa, quindi insomma, fare tutte le settimane avanti e indietro diventava problematico. E poi qui a Pisa avevamo già presi degli accordi anni fa con Roberto Cingolani per prendere poi il coordinamento del centro, di fare il pendolare, ed essendo coordinatore diventava un po' problematico.
Sì. Ecco, tra l'altro voi continuate ad avere 2 centri a Pisa.
Sì, uno a Pisa e uno a Pontedera. Sì.
E adesso che francamente, pur nel rispetto, che sono entrambi ottimi come qualità di ricerca, averne 2 in un posto così piccolo...
Lo so, ma infatti, cioè, io quasi quasi ci speravo anche nella fusione, perché ci permetteva di risparmiare tanti soldi di affitto, che poi affitto non si può chiamare, ma è affitto, e permetteva di unire le sinergie, anche perché facciamo cose che, se vai a vedere, sono molto compatibili l'uno con l'altro. Ora il centro di Pisa sarà prevalentemente focalizzato sul grafene, visto che la coordinatrice è Camilla Coletti. Noi a Pontedera facciamo materiali, c'è il mio gruppo e vabbè, il sottogruppo di Virgilio Mattoli, che comunque rimane con noi, e Mauro Gemmi, che si sposta da Pisa a Pontedera. L'ideale sarebbe stato trovare una stimazione unica per tutti quanti, però vabbè, secondo me ci sono anche un po' di motivazioni anche politiche di mantenere buoni rapporti con entrambe le istituzioni.
Sì, però vedi, io adesso non so come tu sia messo, probabilmente meglio. Io ho subito un taglio mostruoso e vedo che la rete non è nelle priorità di Tifa. No?
Io il taglio non l'ho subito perché più di tagliare di così veramente non potevano tagliare, ero già al minimo.
No, ma infatti, guarda, diciamo la verità, io non è che mi lamenti, cioè io chiaramente ero molto grosso, ma perché all'inizio c'era solo la mia linea di ricerca.
Certo.
Io avevo persone autonome con me, quindi era semplicemente un modo di fare budget, però hanno incominciato a dire che i technology non hanno più budget, da noi il technology è 0 budget.
Come Virgilio?
Anche quello, voglio dire, io ho discusso a lungo con Libero e un pochino con Giorgio, perché io ho detto, scusa, ma a queste persone è stata detta una cosa diversa, no? Cioè, io posso capire da adesso t uguale a 0 in avanti, il technologist è quella figura lì, va bene, ma noi abbiamo preso le persone, abbiamo fatto promesse, le abbiamo a certe condizioni, e poi non puoi andare a dire no, adesso questo qui è tutt'altro.
No, guarda, io ci sono un po' abituato oramai, ci ho fatto il callo, ma ne ho prese di testate nel muro, perché vengono definite alcune cose, promesse, fra virgolette, alcune cose, e poi vengono disattese. Cioè, sicuramente avranno dei constraint dovuti al budget, però, insomma, a volte le persone ci rimangono anche male.
Sì, assolutamente. Rischi poi che queste persone dicano, vabbè, io sto lì. Poi non succede perché abbiamo persone brave, però brave vengono e si danno da fare. Però ecco, già come inizio, e poi io vedo anche la differenza di investimenti, insomma, tra la sede centrale e noi. E poi ti dico, oltre a tagliare, io razionalizzerei, per cui per esempio ci sono 2 centri a Milano, bisogna parlarne, capire, ci sono 2 centri a Pisa, avrebbe senso.
Certo, ma soprattutto qui a Pisa, dove ti dico, siamo tutti e due nanomaterials e le sinergie ci sarebbero.
Sì, infatti. Boh, vabbè, no, capisco che poi vuol dire, non voglio fare le cose troppo facili, no? Un conto, sai, parlare così è un conto farle veramente.
No, infatti, sicuramente ci sono delle ragioni che vanno al di là della mia comprensione, anche perché io sono relativamente giovane in questo ambiente, magari tutte le dinamiche non le percepisco neppure, però, cioè, visto dal basso, diciamo, perché mi considero ancora dal basso, ci sono certe cose che creano un attimino di difficoltà, però dai, non ci lamentiamo sempre.
No, rimane sempre un posto privilegiato IT, su questo no.
Sì, ed è uno dei motivi per cui io ho lasciato il Politecnico poi alla fine, perché sicuramente è un'oasi più felice rispetto ad altre realtà italiane.
L'unica cosa che posso dirti da vecchio è che, ecco, quando invecchi, insegnare ti piace di più, mediamente, ed è una exit più chiara, no? Perché un problema che noi abbiamo in IT, secondo me, è l'invecchiamento delle persone.
Sì.
Cioè, garantire una carriera, una posizione e una gratificazione a tutti, che non è facile invecchiando, perché poi le motivazioni cambiano, sai, la voglia e anche le capacità di fare ricerca cambiano.
Certo, ci sono delle evoluzioni. E comunque quello a me è dispiaciuto un po' lasciare l'insegnamento. Ora, devo essere onesto, le 120 ore erano pesanti, però passare a zero un po' mi è dispiaciuto. Infatti cerco di fare magari qualche corso per i dottorandi, al corso di dottorato del Sant'Anna, dove noi abbiamo i dottorandi. Ora vado 2 mesi appunto a Tokyo, vado a fare visiting professor, quindi terrò un corso. Cerco comunque di mantenere anche l'attività didattica attiva, anche se al minimo.
Sì, no, lo capisco, infatti come fai bene, magari in futuro riesci ad avere un posto, magari via Pisa.
Vedremo quello che ci riserva il futuro, dai.
All right.
Mancano ancora 5 minuti. Io mi prendo
sì, ho rubato la pausa, poi scusa.
No, figurati, mi prendo un caffè, faccio una pausa in bagno e torno. Grazie di nuovo.
Okay, ladies and gentlemen, it is 6:25 in Australia, so I think we can start the second part of our workshop. And we will start with the presentation from Professor Lanzani, and he will be talking about new light actuators for cell opto-stimulation. Over to you, Professor Lanzani.
Thank you very much, Wojtek, for the introduction. First of all, I want to thank you for organizing this. I'm sorry we cannot meet in person, of course, but it still is nice to have the possibility to talk each other, to present the data. And I want to apologize because I have a tight schedule today. Unfortunately, my teaching semester is running, and so I have to fly away immediately, and I came last minute. I'm sorry for this. I mean, I hate to do this, but there's no choice. Anyway, so thank you for having me here. So the title, so I'm going to talk about nano actuator. I start introducing organic semiconductors. I'm sure you know them. Anyway, just to remind, these are semiconductor with very low mobility, but with other characteristics like solubility and color tunability, which are based on carbon. So in this sense, they are also bioaffine. So they are very similar to biomolecule. It's carbon and hydrogen mainly. And these are a very successful story of application in technology. And here you see a few products. One very common is the OLED display in cell phone. So those materials are developed for application. Now, in particular, I've been working with the prototype, prototypical sample, which is polyfluoxyl thiophene. You see here the monomer. Let's see if I can turn on the laser pointer. Okay, this is the monomer of the polymer. And this is the absorption spectrum. Now, you see, comparing on your right, that the absorption spectrum is pretty much overlapping the, say, red and green cones of the retina, the photoreceptor in the retina. And this is actually one thing that will drive the application I'm going to talk about. Now, fabrication, what we do, so we fabricate nanoparticles of this polymer in a very simple way, which is the reprecipitation method. So essentially, the polymer is hydrophobic. You can dissolve it in THF, so in several solvents. Then adding water, as you know, the polymer chains escape the water, so they start to aggregate. And they form spherical aggregates, nanoparticle, that can be selected by centrifugation and dialysis to get rid of the solvent and select the site. And you see below the dynamic light scattering plot, and this typical value of the diameter is between 200 and 400 nanometers, the one that we use. This is an example. Sometimes we also make larger particles. The absorption, then this is the luminescence spectrum. The absorption spectrum I'm going to comment later here. So the absorption spectrum of the nanoparticle tell us a number of things. So we clearly distinguish two components. One is a structured absorption, in which you see some vibrational replica, and that we assign to the crystalline phase. So in particular, this is due to an H-type, say, a molecular exciton. So the wave function is delocalized on the aggregate, and the spectrum is more resolved. The high energy part of the absorption is assigned to the amorphous region, and this is a region in which the chains are not forming any crystalline aggregate. Then there is a tail, which is more questionable, the origin. It sometimes is more evident, sometimes less, and we assign these to charge transfer states, or states that are polarized by the environment, so the energy is a bit lower. Now, spectroscopy doesn't tell you where the components are. I have a guess, actually, that is this. So the crystalline phase is inside, it's like a core, and then there is a shell of amorphous polymer. And then grazing incident wide angle X-ray spectroscopy actually gave me a pretty consistent response. Indeed, it's not just one, but there are a number of crystalline domains inside the nanoparticle. The size is about 10 nanometer, and those are embedded in the amorphous matrix. This is the photoluminescence. Of course, these particles are relatively large, and the nature of the wave function is such that there is no quantum confinement. So you should not think to quantum dot as semiconductor or metal dot. So these are essentially molecular states. Still, so the luminescence is usually coming, the emission is coming from the lower energy states. What is interesting, however, here is that when we go in vivo in living, say, cells, living cells, say cells that are alive in vitro, but sorry, so it is in vitro, but in living cells, you see that there is this absorption peak which disappears, and also the lifetime become very fast when we are in the cell, inside the cell. And this is associated to the emission from the crystalline phase. So that's an interesting observation. I might comment again later. All right. That the first thing we did a few years ago was to study these nanoparticles in a very primitive, simple animal, that is Hydra vulgaris. So here I was inspired by work done by Liberato Manna in our institute, who was testing inorganic nanorods, I think, at that time. So we took the same animal as a simple in vivo test. And the problem here is that I'm a physicist, so I'm a reductionist, but biology is holistic, and so it's very difficult to reduce to do an experimental crucis to reduce to a well-defined system. So here, I mean, if you study a cell, it's different than this little animal, but I discovered later this is also different from more complex animal organisms. Anyway, so we took this little polyps. It's a freshwater animal, and essentially it's a living tissue with some neuron and there's no organs inside. So it's very simple. Here you see that the nanoparticles added in water go inside, so there is a featured uptake of nanoparticle, especially they go in the tentacle, but they are pretty much everywhere. And interestingly, during reproduction, which is by budding, the nanoparticles get dilute, so they go from the mom to the daughter. And reproduction is actually a real copy, so it's not I mean, the remain is by budding. So the reproduction rate is a very good test of the health of the animal. And you see here that untreated or treated with our nanoparticles, the behavior is the same. So our nanoparticles are not toxic and do not affect the life of this animal. What happened, however, is that when we shine light, we see a different behavior and an average different configuration. So this is a contraction rate, so the number of contraction in time, and you see that under light, the contraction rate is increased. And the other thing is that the average conformation is changing. So this, without going into detail of the biology, which I'm not able and perhaps I'm not even interested, it tells us something that is very important. It tells us that the nanoparticle is somehow interacting with the animal. Now, it's not like there's nothing going on. You put the particle and the animal does not react. You shine light and the animal reacts. So there is a light-mediated biological response. Then the biologists we were collaborating with did an interesting experiment, which is based on quantitative reverse transcription polymerase chain reaction. I can barely say the name, I don't know what it is, but I understand it's a way to test some genetic property. Here you see what we did. We were, made some ambient equilibration, then shining light, and then measuring what happened later. And we were testing the expression of one particular protein. Actually, we did with several, but that's the result I want to talk to you. Is this protein here, which is opsin. Now, opsin are proteins that are associated to photoreceptors. And what you see here is that there is an overexpression of opsin when we shine light. And so now what this means is it can be discussed, and I know biologists, so I have a discussion with some of the, I think I'm now here listening, what is the meaning in biology. But for me, say, let's take the message here is not only the animal react to light and behave in a different way, but there is also a deep root change in the biology, because in this kind of genetics. Now, once we see that these nanoparticles can interact, we move on and we take into consideration a different problem. This is the retina. And as you know, the retina has a photoreceptor at the bottom. And then there are layers of neurons, bipolar cell, horizontal Mueller cell, and the ganglion cell, which then connect to the optic nerve, and then the optic nerve go to the brain. We have 120 million rods, which, and say about a few million, 5-6 million cones. And you see here the cones are specialized for the three different colors, white, green, and blue, while the rods are essentially one type, so they see in black and white, and those have very high sensitivity. These are the photoreceptors we use in dark, dim light, and those are the ones that we use in the strong light. Now, the degradation of photoreceptor may lead to a severe disorder and blindness. One of this disease of degradation due to degradation of photoreceptor is age-related macular degeneration. This causes blindness, and it's about, say, 10% of the blind people is affected by this. It's typically for old people, so older than 80, it become very frequent. And it is the estimated number of people having this is 14 million, because it is attacking force. So the degradation starts in the cone, which are in the fovea, which is the area of the high resolution vision. The patients start to lose vision in the central field of view, and then eventually this is enlarged. Now, another disease which is similar, because it is again due to degradation of photoreceptor, but it has a totally different development, is retinitis pigmentosa. This is a genetic disease that can be inherited, and it starts attacking the rods. So it's before the patient loses the peripheral vision. At some point, they remain with a so-called tunnel vision, because it's where the cones are, which are the last to be degraded. At some point, when the disease goes ahead, they become totally blind again. This is considered a rare disease, and actually the estimated number of people affected by this disease is in the order of 1.5 million. Now, for both these situations, when you are at the last step, there is no healing, there are no drugs or pharmaceutical aid except prosthesis. So our idea here was to help restoring photoreceptor by introducing in the eye a photovoltaic material which is based on our polymer. But the reasoning is, the idea is that our polymers are carbon-based molecules essentially, so they are similar to biomolecule. It's a soft material interfacing another soft material, and it supports electronic currents, transport, but also an ionic current. So there is not only mechanical compatibility, there is also some functional compatibility in a sense. What we did was to apply a surgical technique that we already tested before for a simpler type of prosthesis, and this was done by an eye surgeon that is expert with retina, which is Dr. Grazia Bertile, and we did this operation on rats, so lab animals that are affected by retinitis pigmentosa because they are genetically modified. So you see, the idea is that when the photoreceptor degrades, so they disappear. So essentially, when the disease is relatively ahead, you don't distinguish anymore the photoreceptor. And then we come with a subretinal injection. This is a cartoon, but and these lead to the distribution of the nanoparticle. Now, our test, for instance, with OCT done by Maurizio Mete and some histological measurements, so we see that the nanoparticle distribute during the injection because of the viscoelastic liquid that we use also in addition to the water, so they're injected. It's a suspension of nanoparticle. Those are injected and they distribute all over the retina. Then the other thing that we see is that the retina is not damaged by the operation, and they stay there for a relatively long time. Now, how do you test if an animal like this see light, so is rescuing light sensitivity? Well, one thing very simple is to measure the pupillary reflex. And here you see an example before and after light, and here you see the result plotted for 4 different kind of animals, say of samples, cohort of animals. It's about between 10 and 20 animals for each population here, cohort. You see, we have the blue is the wild type, so it's the healthy animal. The green is the animal, a Royal College of Surgeons is the producer, is the animal affected by retinitis pigmentosa. In orange are animals that are operated, so the same surgery, the same procedure, but we are using glass nanoparticles, and which are not absorbing light. And the last in red is the animals that have been operated and in which we inject our nanoparticle. And you see that after 1 month post-operation, the animal, so is 4 months old, and it is developing an important, you see here that is losing the light sensitivity already. When we inject the particle, this sensitivity, light sensitivity is enhanced. And even better if we wait 8 months. So that's a very nice result, because after 8 months, the difference is still there. And you see that the genetically modified animal is almost, say, that doesn't have a real answer, is the response. You see this also flat, doesn't depend on intensity, while the operated one is matching the healthy one, the wild type. And it's something to add, because people is always asking, is why there is still an answer anyway, because you see here that there is a 25% average constriction. Well, the reason is that there are not only retina photoreceptors, there are also other photoreceptors in the eyes. These are melanopsins that are not affected by the disease, and they do respond to the pupillary constriction, to the pupillary. So they are involved in the pupillary constriction. So for this reason, there is an answer to, there is a response also in the so-called blind animal. But of course, you see the difference. Now, another thing we did was a test in the dark and light box, and you put the animal in the light, and you see what happens. And you see that in 20 seconds average, the healthy animal goes to the dark because they don't like to stay in light. But of course, if the animal is blind, like this, or is operated with glass nanoparticle, it's not doing this. And we only recover a quick escape to dark when we operate, when we measure with animals which are injected with our nanoparticles. And again, this is confirmed after 8 months. So it seems that the effect remains for at least 8 months. That's another interesting experiment. This is done, noted, all these experiments are done by different people. And this is positron emission spectroscopy. This is a way to see the activity of the brain, measuring glucose. So I don't enter in the detail that are fundamentally I don't know. You see the healthy animal, and you see that the visual cortex, which is involved, these two shaded area here, is bright in this experiment, but it becomes dark in this experiment on the blind animal. The same thing is it remain like this when we look at the operator with glass, but we recover the bright area when we look at the animal operated with nanoparticles. So this is very interesting because it's a difficult experiment, but it's pointing to the activity of the brain. Last but not least is the real ability to see details, because what I show you now is light sensitivity. It's not the ability to distinguish details. Actually, to what we call visual acuity is what we measure in the optometrist shop. So we go there when we buy glasses, spectacles, and we read letters. Of course, here we cannot read letters, so that the animal doesn't read, but we can show a pattern of stimulus, say pattern of dark and light stripes, and measuring the visual evoked potential from the brain. So as long as the eye distinguish and the brain say that there is the animal distinguish the pattern, there is a signal from the cortex. When this pattern becomes too thin, so there are too many lines to narrow to thin, then the visual evoked potential goes to 0. This cut here, the space frequency at which the visual evoked potential is 0, define visual acuity. Now, for these animals, the visual acuity is something around 1, which is very bad, actually. For a human, it is about 30. So you understand, these are nocturnal animals that cannot see very well, but still is what we have in the lab. So we did the experiment and we test the ability of our operated animal to see these details. So we measure the visual acuity. Here you see the development of the disease and the loss of visual acuity during time. You see that at month 3, when we do the operation, the visual acuity is already less than half, and at month 4 is about 30%. At month 11, when we do the final measurement, 8 months post-operation, it's almost 0. So we are measuring animals that are completely blind. Here you see the situation 1 month post-operation. This is the genetically modified animal. And now, again, you see that with our nanoparticle, we can rescue visual acuity almost completely. So we are able to rescue vision. So these are blind animals that recover vision. And with some scattering, some more noise, it's interesting to see that even after 8 months, so with an animal that is 11 months old, these of course are difficult experiments because the old animal does not support anesthesia very well. You see that we are able to recover, to rescue visual acuity. So the nanoparticle recover visual acuity. At this point, the final to conclude, what is the mechanism? Because that's interesting from being a physicist. I can imagine 4 different mechanisms. One is capacitive. The accumulation of charge generates some change in the charge distribution and is affecting the cell attached to the nanoparticle. The other one is paradigm. There is an electrode and a counter electrode. So when we shine light, we generate currents. Now, this is what happened with silicon, but it requires a level of current, which is in the order of kiloamp per square centimeter, which is very high for our situation. There is the thermal effect, which is certainly occurring. When we shine light on a semiconductor is absorbing, the temperature locally is increased. However, in order to do this, you need a certain power, which usually we don't have on the retina. And then there is what I call chemical or photocatalytic, if you want. not only the charge is accumulated, but there is also some chemical reaction on the surface. Now, what I think is that when we shine light on the polymer, there is accumulation of charges. And here you see essentially why. This is a simulation in which we solve the Poisson equation and the charge transport and recombination equation. So there is the diffusion and Poisson equation together, the drift diffusion equation model. And you see the light come from this side. And what I'm considering is expressed in this cartoon here, that holes have a relatively larger mobility, is small, but it's larger than electrons, while electrons are very, very slow. Essentially, they don't move. So what happens when you shine light? Well, you see in time, you saw the development in time, the holes are free to distribute homogeneously due to diffusion. And the Coulomb attraction, that's what our simulation is telling us, because there is a certain screen, so the Coulomb attraction is not enough to stop, to hamper this. So the holes distribute homogeneously in space, the electrons essentially do not move, and these generate an electrical polarization. You see here, blue is negative charge, red is positive charge. So when you shine light, there is an electrical polarization, and the surface that is exposed to light become negatively charged. Okay, now this, including also adding the order of magnitude of the light that is reaching the retina, if you want, then I have some supporting slides for you, is suggesting me that the two possible mechanisms are these two, the capacity of the chemical. I'm over, yeah, the time is, so I'm finishing. So, and it is indeed what, so both together are probably what is going on, no? Because we have accumulation of negative charge, which interact with oxygen, which is helping in the long-lived accumulation of negative charge. All right, to do this experiment, a lot of people is involved, and you see here a very long list of people. That the major partner in crimes are the neuroscientist Fabio Benfenati and the eye surgeon Grazia Pertile. And the last thing I want to say is that we launched a startup, it's called NovaVido, because we want to bring this technology to human. And thank you very much for your attention.
Fantastic, really great presentation, and congratulations on a startup company. This is
Thank
really
you.
exciting, yeah. Very good.
That was hard. I think it was harder than doing the experiment, but at the end we did.
Oh, it's absolutely stunning. Any question?
I see.
Maybe
Jada.
I'll start actually. Can I ask you, is it concentration dependent, your effect as well? So let's say if you increase the concentration of nanoparticles, would you have a stronger effect?
Mm-hmm. Good point. Say the answer is, I don't know. In vivo, we never measured this because we start with a certain suspension and we were happy of the results. Probably we were lucky. The thing is that to do this experiment is very difficult. So you cannot change too many things. What we see is that the nanoparticles tend to aggregate. So they form aggregates of about 1 micron size. And that tells us that probably if you change the concentration, there is a certain range in which nothing changed because you do the same kind of aggregation. What I know in vitro is that if you get too large aggregates, they tend to be toxic. So if you make large patch of polymer attached to the membrane of the cell, that's not very healthy, actually.
I have another question, but Chiara has the hand up. So Chiara, do you want to ask a question, or it was accidental?
No, thank you. Actually, I would like to make a few questions. Thanks,
Yes,
first of all, for the very nice talk, and because it brings really hope for the restoration of eyesight in humans. And my questions are mostly dealing with a Hydra model. They can be considered as a curiosity, but I was curious to hear a little bit about the light responsiveness of these animals, because you mentioned that you could observe a change in behavior after interaction with nanoparticles. But did your nanoparticles promote this behavior, or was it a gain, so sort of gain of function, acquisition of a new behavior responsiveness from these animals?
Mm-hmm. No, okay, so that's a good point. And I remember, if Degli Innocenti is here, I think he might remember, and maybe also Gianni was involved, we had a discussion on this. Is this a function gain or a function loss? My intuition, but probably is wrong because I'm not a biologist, was a function gain. Anyway, let's first say what happened. You put the nanoparticle and you don't see anything. So the animal behave exactly as not having it. When you shine light, you see differences. So they start to move more, and the average size is different. So then you see a difference which is light-induced. It's always spot-induced. And it's only seen when there are nanoparticles. So if you shine light, the same light on the hydra without nanoparticle, they do not react. I think also depends on the strand of the animal, but what we had was essentially light insensitive. Now, I was thinking function gain. I was saying, okay, the animal thinks there are more photoreceptors and it is making more opsin to cope with this photoreceptor. But then biologists told me, no, it could be the other way around. It could be a function loss, actually. So the animal feel like something is not working properly because the nanoparticles are somehow interfering with the path, and then it is generating new opsin to substitute, to correct this. At the end, I don't know what is the truth. But for me, I mean, I was showing this because historically, this the first thing we did, is I think it's a demonstration that something is occurring, and it tells us that there is a lot to be understood in this soft material interface.
Thank you. Very interesting point. My second question is...
Sorry, Giada, before you go on.
Sorry.
You know, just because if I may recall the previous discussion with Guglielmo, maybe I can add something just very shortly, and then I leave you the audience. So I indeed, Guglielmo, what I wanted to say is that it's not just about gain or loss of function. I think the most important thing to notice here is that the increase that you notice in opsin production occurs at a cell level. So it's a cell-by-cell reaction that each of them, so each cell basically has to adjust to something that is missing. This is my point. If things were like you were saying before, you would have this on a more systemic scale, if I may say. So since a cell senses a kind of a lack of light, it reacts homeostatically by increasing opsin production. So that would point to a loss of function. This is my point, just to recall the previous discussion we had. Yeah, sorry for interrupting.
Yeah, no, thanks, Andreas. Yeah, I remember that we had this discussion together. Okay, so.
Sorry, Gianna, if you don't mind, because Teresa has also a question. So maybe we'll go Teresa first, and we are a little bit behind the schedule. So maybe you can ask President Lanzani after the talk. So
okay,
Teresa.
Thank you.
Thank you.
No, thank you, Guglielmo. Very interesting, of course, to see how it's evolving. When you talk about the model, the fact that the electrons are moving less than the positive charge is due to the fact that the particles, let's say the polyelectrophene, are the negative one, and they are in some way in the matrix. So is the counter electrolyte the positive one that they can move, while they are, let's say, a little bit more fixed within the cornea, right? I mean, the layer where you inject them.
Well, no, it's a property of the organic semiconductors in general, the solid state of the organic polymer. The electron is essentially trapped by oxygen or other, by the polymer itself. So it behaves like a recombination site, and it has this very low mobility because they are trapped. So that's the reason why these semiconductors are most of the time p-type. It's very difficult to make an n-type organic semiconductor. There are, but it's difficult to make. So it's a fundamental property of an organic semiconductor film or solid state. Then the particle is like it's a small chunk of solid state. So it's a condensed phase, and it is behaving in the same way. So it's a property.
Okay, let me reformulate. Do you think that in addition to that, even the fact that you are talking about counter-ion, the positive one, that they move quicker, has something to do? Have you tried particles that, and the other way around, are positive with counter-ions that are negative?
No, yeah, good point. This is interesting. No, I'm trying now. We're using perovskites because there you can. I mean, we know here how to make devices, and we can change this polarity if you want. But I still don't have any results. So I could never test with the polymer or with the nanoparticle test the opposite behavior. So I don't know. Okay, that's a good point. My feeling is that whatever, if you have a charge appearing on the surface, is probably felt by the cell nearby. But that's because you see also in neuron, the interface is very tight between the nanoparticle and the cells. These are neurons in vitro, but you see that the membrane tends to stay around, and so they feel the charge that you accumulate there. But that's my, I mean, maybe I'm wrong.
Okay, thanks.
Excellent. Thank you so much. I think we'll have to stop here and move to our next presentation. So thank you so much and congratulations on the fantastic results.
My pleasure. Now, apology again. It might be that I disappear and come back, unfortunately, because I have people coming, but I'm sorry for this. I mean, I hope that if there are questions, then I'm very happy to receive emails and to talk if someone wants to ask. Apology really for my small appearance. Short appearance. Thank you very much again for having me.
Thank you so much. Thank you.
Bye-bye.
Thank you. So now we will move to a talk from the University of Sydney, and the speaker is Nick Hunt, and he will be talking about a NanoDeLiver. Nick, over to you.
And I've muted myself. All right. Hopefully that is on the right screen for everybody.
Yes, this is correct. And awesome. We can see and hear you.
Awesome. First of all, thank you so much, Wojciech, for inviting me. The scale and size of these presentations has been amazing, and I'm in awe and honored to actually even be here and be part of this session. The impressiveness of the work is truly fantastic. So the work I'm going to talk to you today is something we've been working on the last couple of years, and we're slowly developing using nanotechnologies to target the liver. As you all know, nanotechnology readily goes to the liver, and it's not so much of a difficult option to target, but to get precision delivery and uptake and drug regulation potentially can be. So I'm from a more different background to most people here. I'm based at our clinical school and at one of our hospitals here in Sydney in Concord. And we work on a multidisciplinary approach, much like a lot of our previous speakers. And we're always trying to solve a clinical problem that we see both in the clinic and within the hospital. And we work not only in our hospital setting, but also Sydney Nano, that's helped facilitate this particular meeting, and also the Charles Perkins Centre, which focuses on trying to solve critical problems for diabetes and obesity. And the liver is a fantastic target for particularly for diabetic conditions and also for obesity, because it's the metabolic hub of the body. So nanotechnology has such a unique opportunity to be able to target it. As most of you all would know, conventional nanoparticles are rapidly cleared from the body within minutes, accumulating in the liver, and then can be cleared out either by kidney or excretion. So what we've originally got is using conventional nanomaterials, quantum dots, organic nanoparticles, and organic polymers. And then we've looked at, can we develop therapeutics for type 1 or type 2 diabetes, metabolic disorders, and liver diseases. And we're going to talk about some of the publications we've put out more recently and some of our more interesting work. So ideally what we want to look at is medications for diabetes. So metformin, and insulin, GLP-1 agonists, and then start looking towards what else you could do in terms of immune regulation. And that's going to be the basis of what I want to talk to you about today. Just as a brief overview for anyone who's not 100% up with nanomaterials, what I'm going to be talking about is quantum dots, so very nanoscale type materials, 5 to 6 nanometers originally when they're originally synthesized. And I probably won't touch on carbon quantum dots, but we're also going to look into this particular type of area. And what we're going to be looking at is looking at different targeting strategies for different cells that exist within the liver, how you can control release of different drugs and proteins, how you can protect them from lysosomal degradation, and how you regulate endocytosis. Just as a bit of a brief overview of different techniques that can be used to visualize nanoparticles, TEM, and also their functional chemistry of FTIR. Just in the video here, I want you to just kind of keep, while it's running, watch it interact with the cell, and also watch what happens internally within the cell as well. So what this is an immortal cell line of the liver sinusoidal endothelium. So what was highlighted really particularly well by women, by a group in Canada a couple of years ago, was that there's multiple pathways that inhibit nanoparticle survival in systemic circulation. So these can be mononuclear cells, and also given a liver sinusoidal endothelial cell. So this is an immortal cell line of the LSEC. And what we're looking at here is nanoparticles that have been mixed with human serum to create a protein corona and see their interaction with the cell. And what you can see is this large clumps of nanoparticles interacting with the cell that are taken up into a lysosome in large groups and then become monodispersed as they're then released from that lysosome. What we also have looked at is different radio labeling type of techniques to be able to track where our nanoparticles go within our body. The example I only want to give here to focus on is we've used radio-labeled metformin, which is a very common type 2 diabetic, and we've attached it to a quantum dot. And the only brief point from this slide is metformin alone has therefore uptake into explants of small intestine. You could rapidly improve that with attachment just to a quantum dot using the same radio label. And what also occurs is metformin is blocked by a particular inhibitor that can be given. So they're blocked by protein pump inhibitors. And using nanotechnology, you can mitigate that effect. So it no longer is blocked by a particular protein pump inhibitor. It can be blocked by an endocytosis pathway that's more broadly used, such as soluble ligand channels or soluble anion channels that readily take up soluble nanoparticles that are less than 100 nanometers. It really doesn't come as much of a shock for most people of if you give animals and in vivo models nanoparticles, they'll rapidly accumulate in the liver and then they'll be cleared. What we have looked at is what happens when you give nanoparticles orally. And they do show rapid translocation of this across the small intestine into the mesenteric veins and then pass through the liver, but they're cleared by the liver, so they don't reach systemic circulation at all. At 30 minutes, within 2 hours, within 24 hours, there's no systemic circulation from orally ingested quantum dots that are made of silver sulfide. They go into the liver, they're translocated to the bile and then cleared back into the small intestine. What we wanted to do and focus on is if you've got this mass accumulation that's accumulating in the liver, can you reach certain different cell types that are occurring there? So the first cell types that are going to be taken up by are going to be your Kupffer cells, they're your MPS. Then there's going to be your scavenging endothelium, such as your LSEC, and then they're going to pass to the hepatocytes where they can be recycled into the bile. So liver sinusoidal endothelium have a few tactics to facilitate nanoparticle clearance and removal. They can be passively removed by fenestrated surface. They can be cleared by transcytosis, and they can be cleared by cellular scavenging pathways. All of those are regulated by different subcellular and surface receptor channels, and you can target those different ones with different surface proteins. So if we've got a quantum dot, we've radio labeled, and we attach a molecule such as FSA, which very readily binds to a scavenging receptor on the liver endothelium, you can target the endothelium in preference to the MPS. If you wanted to use heparin, it promotes transcytosis in LSEC, so it doesn't retain these cells. It isn't picked up by mononucleosides, but it's very quickly cleared by hepatocytes in the bile. And that presents opportunities of if you can target one single cell type, you can develop therapeutics that can go to those particular cells. So that's kind of where we're kind of going from this. What we also wanted to confirm within an in vivo model is what we saw in our cell culture model previously of having nanoparticles suspended in a protein corona, interacting with a cell membrane, and then becoming monodispersed within a liver endothelial cells, confirming that what we were seeing before in vitro can occur in vivo. And if it can be monodispersed, you can have release of materials when it's in that particular state. Also, that the cells didn't show any particular immune response, no aggregation to them. They got taken up to the lysosomes, but then they can be released, suggesting there is different pathways you can act on. So what we've done first of all is simple coatings of different drugs onto nanoparticles. So effectively, they look like a nanoformulation, just with surface coatings of nanomaterials. So we're not using any polymers at this point. They look, according to FGR, like the drug themselves. So these are metformin-coated quantum dots and nicotinamide mononucleotide quantum dots. And there's a certain amount of drug you can attach to each one of them, but we wanted to see if you can give them orally and they can be cleared by the liver, can they deliver a drug there before they're removed into the small intestine and the feces? And the answer we observed was yes. Metformin has an incredible ability to allow glucose sensitivity, and it does it in a dose-dependent way. So if we give a mouse some glucose, he'll have a spike in its blood sugar and it will slowly drop off over time. And if you pre-treat it with metformin, it has a dose-dependent effect on blunting that curve. And then we measure the area under this curve to measure the effect size. What we saw with the quantum dot formulation is you could have exactly the same effect in a dose-dependent way, but with a much lower dosage that needed to be used. So you can shift the drug response curve to the left to lower the amount of drug that needs to be given. Whilst metformin is not particularly toxic, it is a great proof of principle demonstration. What we also saw is that it could have a much more faster uptake and effect, suggesting that even though we are using much lower amounts of drug, it can still be beneficial in a much shorter period of time. And that becomes really important when we move forward later to start talking about insulin. And the last thing I do want to mention for metformin is it's dependent on a very specific uptake channel, and that's an OCT1 channel. This has clinical implications for type 2 diabetics as they develop desensitivity to this channel. And there's a proportion of people who have type 2 diabetes that you can't actually treat with metformin, which is your first line treatment. What we've done with our quantum dot formulation is not only increase the amount of uptake you can get into hepatocytes, but we've got rid of the blocking effect of osmopriol, which blocks the protein pump inhibitor, and switched it to something else. And that means we can mitigate that loss of function for the OCT1 channel in a potentially for a patient. We've also looked at another drug, NMN, to see that the channels that are responsible for facilitating its uptake can also be blocked as well. So we've looked initially at metformin, which is our first line treatment for type 2 diabetes. And what we wanted to do is look at the second line treatment for type 2, which is insulin, but also highly critical for type 1 diabetics. Type 1 diabetics are unable to produce their own insulin due to an autoimmune response that degrades both the insulin producing cells in the pancreas and also can recognize insulin itself as well. So what we're designing here is if we can have an orally administered quantum dot formulation with a protein attached and then coated in a protected polymer, can we then use this to try and address the three critical problems for an oral formulation? That is that an oral formulation needs to replace injectables based on how much fear and problems with compliance. So if you can replace it with a tablet, that is the most ideal solution. The next problem with an oral formulation of insulin is timing. So it needs to work within the same time frame that insulin would work. So that's 15 minutes, which would be an amazing achievement for an oral formulation. And the last thing is safety. Drops in blood sugar is a very common event with type 1 diabetics, resulting in hospitalizations for them. At least 1 in 100 a year will have incidents of hypoglycemia, require hospitalization and treatment. So you want a formulation that's safe. And what we've aimed to do is address those three criteria. So what we're presenting here is wild type mouse data. They're given their subcutaneous injection of insulin or an oral gavage of insulin at the same time. Then they're given glucose to stimulate an increased surge in blood glucose and seeing how fast insulin can clear the glucose that's been given. So we had a dose similar effect based on 2 units of subcut insulin versus 20 units of our oral insulin formulation. And we also saw with the subcut, you can get dose-dependent effect. And with our oral insulin effect, it did have a dose-dependent effect, but then it petered off. What I do want to highlight about this is if you give a dose-dependent effect with subcut insulin, you're going to induce hypoglycemia. What we saw with our oral formulation of insulin is you could flatten the curve, but you couldn't induce hypoglycemia in these animals. So that's the element of safety that needs to be addressed. So you can't induce hypoglycemia, and if you increase the dosages, you still couldn't induce hypoglycemia in these animals. And that was a really exciting area to kind of look into. So what we've then gone and done is proof of concepts in multiple animal models and hive primates. So I'm only presenting a subset of the data here, but this is an oral insulin tolerance test in a diabetic animal model, so a NOD mouse that's diabetic. They're given the subcut injection of insulin or the oral gavage of insulin at the same time. 15 minutes later, they could have the same physiological effect in NOD mice, which is a fantastic achievement with this particular type of formulation. What we then wanted to address was a second animal model, of course, with STZ treated rats. But what we wanted to do was put it in their drinking water, see if they'd naturally take it, and if the natural drinking of water could regulate their blood sugar. So what we did was we gave our animals a fixed dosage, 150 units, which is a very high dosage of insulin. And what we saw is they could achieve a basal level of glucose between 15 and 25, and they can maintain it throughout the whole treatment period. So effectively, they were drinking their own therapeutic, and it wasn't inducing any hypoglycemia, it wasn't inducing negative effects on these animals. And then we had a washout period that showed that it was the therapeutic that was inducing that effect. Lastly, I just wanted to touch on, we then examined hive primate models. So these are baboons that are non-diabetic. And for this, we had to give them a natural tablet, or in this case, it was actually a piece of chocolate we formulated with our nanotech within it. And this could induce a dose-dependent effect in an insulin tolerance test in non-diabetic animals. Within a therapeutic dosage, it should be viable for commercialization and development. Whilst an oral insulin formulation is a fantastic area to kind of work in, the key thing of why we do a lot of these particular medical research strategies is to try and prevent different disease developments. So what we're looking at here is, can we modulate the autoimmune response in NOD animals for type 1 diabetes? So a type 1 diabetic will develop an autoimmune response very earlier on during their somewhere between 5 to 12 years of age. In NOD mice, this occurs about week 8 or week 12, and they develop a autoinsulin antibody that then goes and facilitates activation of CD8 cells that then degrade and destroy beta cells in the pancreas. And what we're trying to focus on here is, if you can target the liver, the other key cells that usually facilitate clearance of nanoparticles are your Mps cells. These are cells that are incredibly important for immune regulation. They correspond with dendritic cells. They can facilitate T cell regulation. And the most prominent therapeutics we currently have and are developing for type 1 diabetes, particularly in immune prevention strategies, are via those strategies. So it presents a really unique opportunity to use nanotechnology to target those cells that usually is the bane of existence for nanotechnology nanotherapies. So what we've done here is we've looked at, can we suppress the autoimmune antibody response? Can we promote upregulation of a regulatory T cell that's designed to, and has been shown in previous studies, to facilitate immune regulation and suppression of type 1 diabetes? And the answer we have so far with the formulations we've developed is yes for both. And now we're looking at, can we develop a dose response effect to that? And can we use different therapeutic dosages and different time points to induce it? So usually NOD mice will start developing diabetes around week 12, and by week 25, 26, you'll usually develop diabetes in all of your NOD female mice. What we've shown here is the beginnings of a dose-dependent effect with a lower dosage, a medium dosage, and then a high dosage, which is coming through here. But the interesting component is the one that's at the top. So this is the one where we only give singular treatments during an optimal window that we can identify based on their climbing autoinsulin antibody, to do a single one-off weekly treatment and then observe that they don't develop diabetes later on. So it's really exciting to see if this continues along, and just the potential for nanotechnology for immune regulatory therapy. And that's particularly what I'd like to facilitate engagement in this workshop to be able to do, since immune regulation is a really critical area, and nanotechnology has so much potential for it. The last thing I did want to touch on, and no one's told me I'm running out of time yet, which is really good, is the concepts of aging. So from a geriatric clinical point of view, if you've got an elderly patient, so if you're over 60, 85% of people over 60 will be on one medication. 50% of them will be on 4 or 5. So who we're developing therapeutics for largely is going to be an elderly population, and they do demonstrate quite different physiology than young animal models we might be using to develop therapeutics, and we need to take those considerations into account. For instance, Kupffer cells, the main Mps cell, will be more readily activated in older patients. So the immune response, the immunoglobulins, the complement that's going to be available to bind to nanotechnologies is going to be heightened, particularly in the patients that you're trying to treat. You have loss of the passive filtration that clears nanotechnologies. You have a whole bunch of buildup of ROS, and they can't properly excrete materials. So you're going to get longer circulation of nanomaterials, but there's problematic aspects with these patients that do need to be considered when designing nanotechnology. Nanotechnology, like a lot of medications that we have, will vary with older patients. For instance, there's a relationship that exists.
3 minutes, Nick.
Awesome. There's a relationship that exists between lipid-related type drugs and highly hydrophilic drugs, with lipophilic drugs having more freedom to move around based on the amount of weight that people do accumulate over time, and there's less room for polar drugs. And there's also less potential for affinity binding, so the protein corona between older patients and younger patients can be completely different. And it's less albumin that will be available and more complement, more IgG, and more immune cells that are going to be available. So it's an area that needs to be addressed. But the main point I'm going to bring up from it is those problems are problems for therapeutics as well. So a lot of medications lose their efficacy in older patients, and they develop higher toxicity. And nanotechnology has a really unique platform to try and solve some of those problems for drugs. What I'm showing here in the last section is 2-year-old mice. So these are very old mice, equivalent to about 80 or 90 years in humans. And we've given them an NMN treatment, so nicotinamide mononucleotide, which in younger mice induces fantastic effects in their glucose regulation, insulin tolerance, and insulin resistance. However, at a very high dosage in these old mice, it can induce no improvement in glucose, glucose response, fasting and fed insulin, and their insulin resistance scores. Whilst if you conjugate it and coat it onto a nanoparticle, you can facilitate a nice effect and show that this drug can be effective in a really old animal model. And although both are getting into hepatocytes, they're both promoting the effector molecules you would ideally want, it wasn't having a physiological effect. So there's that extra efficiency that can be promoted just by using nanotechnology to try and address those problems. Just as a last area of kind of direction that we're going into, I should highlight that the oral insulin technology we've also developed it as a protein peptide carrier. So the other proof of concept molecule we have is a GLP-1 agonist. This acts within the pancreas. So the idea is we actually have release of insulin within the liver that then gets delivered to hepatocytes. But with GLP-1 agonists, they don't act on hepatocytes. They need to enter systemic circulation. So we've shown that we can get the same effect size with the GLP-1 agonist as we have with our insulin, but it needs to travel through the systemic circulation and then release in the pancreas. what we're also starting to work on is what else can we deliver with this technology? Given that we can show protection of a protein and a peptide, can we deliver siRNA, microRNA, and can we deliver CRISPR? Since if we can get singular cell type targeting, you can address particular problems that exist within those cell types. And that's an immense area to work on, given that a lot of clotting factors are produced within the liver, particularly within the liver endothelium and hepatocytes. Hemophilia is caused from one cell type, and that's the LSEC. Being able to precisionally target that cell without an AAV vector is a major challenge that we're going to have to start addressing within that space for hematologists. And lastly, we're also looking at targeted delivery for immunoregulation in type 1 diabetes. And of course, there's a lot of people that have contributed to this work. And I'd just like to take time to thank them all, particularly ones from Anzac, from Wojciech as well, who's continued to help me and facilitate a whole bunch of experiments. I'm looking forward to us getting back in the lab and actually being able to do some. As well as Jared from business, who's been a great support, previous students and funding from Australian Diabetes Society, Haemophilia Foundation, Sydney Spark to look at the commercialisation of oral insulin, as well as the NHMRC, which is a major funding source in Australia. And I'm happy to have any questions that you might have. And thank you all for listening.
Thank you so much, Nick. Really, we are a bit behind the schedule. I can allow only for one question. Anyone? Nick, actually, I will ask you because I've never asked this. The slide which you had, the tissue and the distribution of individual particles, do you know what's the mechanism behind that? You have a cluster with corona, and when it's uptaken, is actually broken up into the small individual particles. Do you understand the process behind this?
So we've done this in cell culture. So we've seen, and we go through it, I think, in the 2020 ACS Nano paper, where we have an increase in lysosomal expression with nanoparticles. They get taken up into the lysosome, but after a couple of hours, those lysosomes start breaking apart and the nanoparticles are just released into the cytosol of the cell. What we're also interested in doing and currently developing is, can you release and break apart that lysosome and then get the monodispersion again? And the answer is yes, and there's a lot of lysosomal escape polymers that are around that you can code onto the surface of your nanomaterials to facilitate that. The other thing to point out is you need to facilitate a particular receptor subtype to make sure it's scavenging uptake instead of transcytosis. And distinguishing between those two is really critical to getting different cell type targeting.
Excellent.
I've just kind of highlighted, if anyone would like to heckle me, there's my email.
Okay. Terrific. Thank you so much, Nick, for the presentation. Great work. And we will now move to a presentation from IIT Italy. And Teresa will be presenting on nanoparticles for magnetic hyperthermia and radiotherapy to treat tumors. Over to you, Teresa. 20 minutes.
So thank you very much. I guess you can see the screen. And it is right. It is a great opportunity for me to be here and present the work done over a few years. And we do develop inorganic nanoparticles, and we do assemble them in a different geometry, different structure. So the particles we produce have nanoscale size, 10, 20 nanometers, but then we use to arrange them in different geometries because we can affect the magnetic properties, especially the magnetic hyperthermia. And then we can even put in much bigger containers. And we do study this in vitro and in vivo. Now, let me briefly introduce. Superparamagnetic nanoparticles are very interesting because in absence of magnetic field, even at body temperature, they can have no magnetization, no response to magnets. So they are inert carriers. So you can use them as a carrier. But as soon as you expose them to different fields, they respond. So in case you apply an alternative magnetic field, what happens is that internal and external friction of the particles will make the nanoparticles, let's say, vibrate. And in this process, they accumulate energy. So they accumulate from the magnetic point of view and hysteresis. This energy can be released to the environment, and the way it is released is in the form of the heat. So you can use a high frequency magnetic field to alternating magnetic field to generate the hysteresis. And the heat dissipation power, it depends from the area that you generate. Larger is better the particles heat, and to the time you go through this cycle. So the frequency. Frequency is fixed for application in vivo. So it has to have a specific value. But of course, the properties of the materials generate a larger hysteresis. And the SAR, specific absorption rate, is a way to measure the particles heat, which is given by the area, the frequency, and normalized to the magnetic materials. Now, what is the state of the art of magnetic hyperthermia? Fortunately, we are at the clinical level, so it is used to, nanoparticles are used to be intratumorally injected at glioblastoma, and upon application of an applicator that has been developed by Megforce company, you can locally apply the alternative field, heat up the particles, and do this therapy as a adjuvant therapy for chemotherapy and radiotherapy. The dose of particles so far used is very high. And this is the big problem because they are made also, these particles, to stay at the brain. And because of that, you cannot use magnetic resonance imaging for doing, for monitoring the tumor progression. So in some way, our goal is to look at these studies, clinical trials, and redefine the type of particles in order to have much better heating nanoparticles to reduce the dose, and have particles that can be degraded. So over time, they get removed, and then you can follow the progression of the tumor by MRI, standardly used for brain, and then, of course, to have a more effect, combine heat-mediated drug delivery, so having possibility to deliver drugs locally based on the heat. So targeting is also another aspect that we care about. All these parts have been, let's say, started, are the activities in which I'm involved. So to measure the SAR, we use calorimetric measurements. You have an alternative magnetic field coil. You apply the frequency, which in vivo is 110 kilohertz, and then you record the temperature versus time, and then you measure the specific absorption rate. The type of nanoparticles we have developed are different. Some are monocomponent, like iron oxide. But as you can see, most of them, they have shapes that are far from being roundish. And especially, we are working a lot with the cubic light shape. We can even have materials which are multi-component materials, heterostructure, like in this case, gold and iron oxide. But why the nanocubes heat better and we produce this rather than the spherical one? Because if you look at the particles at the surface, at the nanoscale, surface effects are very important. And the spin counting, which means the spin disorder at the surface, is much higher on a curvature surface at the nanoscale than for a flat surface. This affects very much the properties. So with our nanocubes, just by modeling the cubes, we can obtain much higher magnetization of saturation. And the effort we do, which means that even this, which is a larger, let's say, a larger loop, could affect the heating properties. We have been setting protocols to produce these nanocubes, and we have our own patented protocols in which the other parameters that we control very well, beside the size, is also the monodispersity. So how similar are with each other the particles? And by using this dibenzyl ether squalane mixture of solvent and this heating profile, we can have nanoparticles at different size, controlling the size of nanocubes from 14 to 35 nanometers, but even getting bigger particles which are truncated octahedra. We can transfer because they are coated by surfactant in water with different procedure. We can be quantitative. Usually polyethylene oxide is the best to use for bringing them in, or polyethylene glycol is the best to use to bring them in water. And when we study the SAR properties at conditions that are the frequency in vivo, we can define that the 90 nanometers particles, which are the superparamagnetic one, still have, let's say, a very high heatability, and this SAR is 10 times, 20 times more than particles that are used in vivo. So this is very important. Moreover, when you apply frequency and field, the HF product has a certain biological safety limit. So you can have particles that they have to heat up on this side of this HF factor. If they are above this, they are not really useful for magnetic hyperthermia in clinics. Perhaps they are useful for other applications. When we compare particles with what we have on the market, our nanocubes are very similar to magnetosomes produced by magnetobacterials in terms of heat produced. And it is not the case that they are, let's say, shaped. So they are faceted particles. The point is that the method I presented before, it's a method that allowed to obtain milligram scale particles, but to move in vivo, we needed to have gram scale production. And in the last 3 years, we have been working on procedure to maintain the same quality of properties, but having, and therefore SAR performance, but having much higher heat performance than the standard one. And indeed, we could produce these nanocubes by a solvothermal method, adapting condition than of the thermal decomposition method. And the other way we work to obtain nanoparticles, sorry, here, there is this that I should move, so here it is. You can see that the heat performance of our particles with respect to whatever it is commercially available is by far much higher. Okay, there is another way to increase the heat properties of the particles, which is by assemble them. So we have been studying different types of assembly of particles through polymer, and we have found out that whenever you do clustering in 3D, you reduce the SAR properties, the heatability. But if you make chains of particles, you can then have still nanoparticles that they heat more than the initial one. So having the same dose, but in a chain-like configuration, you can improve the heat of the particles. In this sort of bidimensional arrangement of particles, we see that the heat, the initial heat of the particles is higher, it's similar to the initial particles, but they can be accumulated at the magnet much easier than the single particles. In addition, these particles have been assembled through a polymer produced by bacteria that is esterase cleavable. And when the polymer cleaves, when the enzyme cleaves the polymer, we get these chains. And these chains increase by far the SAR of the particles because they go back to this controlled arrangement of particles. So this is also a very active approach that we have. As I said, we wanted to combine magnetic hyperthermia with drug release in a controlled manner. So we have been working on thermoresponsive polymer, which are polymer that can accommodate a drug within the hydrophilic shell. But then upon increasing the temperature, you go and break the hydrogen bonds. And because of that, the polymer can shrink. And in this process, release the drug that has been previously incorporated. And so we have been over the years developing nanoparticles that can heat up in vivo, because this is the very important concept. And one thing we learned is that we really need to have single particles coated by thermoresponsive shell. And to do that, we have patented this procedure by which, using photoinduced copper-mediated polymerization, we can grow polyethylene glycol methacrylate polymer at the surface of the particles, reaching single particles coating. So if you look at the particles, they have a single polymer shell around, and this we can produce at milligram scale. And if we apply, we can also tune the temperature at which the polymer will shrink. So for instance, we can go to 42 degrees to have the shrinking of the polymer only when we go above body temperature. And then these particles can be heated in multiple cycles and they maintain the heatability. And it can be applied to different sizes particles. And we can load doxorubicin within the polymer and clean up by the extra drug by applying the magnet. And then when we can load 47 microgram of doxo per milligram of iron, and then we can show that by applying clinical condition hyperthermia, we can release, for instance, with the 3 cycle, of hyperthermia, in 90 minutes, 28% of the drug incorporated when we apply the alternative field, while to do this just at body temperature, at 25 degrees, it will take much longer. So it will take 8 days. So you can boost the release. We have been studying this system in vivo for the efficacy. So we develop a xenograft tumor model. We inject the particles, we apply hyperthermia, and this is the hyperthermia system for the animal. And then we study TM, ICP, histopathology, and we follow tumor growth and survival rate. And as you can see, the group that has received the doxo loaded nanocubes and it was exposed to the hyperthermia, so the dual synergy effect, can completely reduce the size of the tumor, while in the animal, let's say, survive even longer in this case. And this is not the case for all the other control groups. What's happening locally is that with respect to the injection of doxo by systemic way, where you can see the doxo located at the vessel. In this case, the particles are more spread when you have the TR cubes nanoparticles. And in addition, the heat induces a much higher mortality of cells. So here there is a necrotic tissue, so no DAPI cell covering, which is an indication of the spreading of the particles along the tumor with the heat, together with the drug release over the tumor, favors the necrotic cell, the death of the cell. Okay, biodistribution of these particles on healthy animals have shown that these particles within 2 weeks already, they are of course accumulated at the liver and spleen, but they can be removed through the kidneys. So, and this is the control mice. And upon 5 months, even the liver is cleared. Already from 4 months, we see removal. This was done, a study in collaboration with Florence Gauzot.
5 minutes.
5 minutes, right? So let me go fast. So we also studied this effect of hyperthermia looking at cancer stem cells, because this population is harder to be treated than fast proliferating cells. So we are developing from patient-derived colorectal cells tumor spheroids, and we do apply the treatment. And we then, I skip this, we then check the effect of hyperthermia on these spheroids, and having a look to the overall population, like in this case, so cytotoxicity to see how the different treatment interacts, and also more specifically, so you can see that indeed you need hyperthermia, but you need also TR doxo, the same materials that I was showing before, so the release of doxo to kill completely the cell, because the heat alone is not enough. And then we follow the tumor, the colony formation assay. And again, you don't have colonies only in the case of TR doxo plus hyperthermia. And we do studies, confocal studies on this stained cancer stem cell. And we can see that indeed the effect of doxo plus nanoparticles and magnetic hyperthermia is the fact that indeed you induce the cell to divide with the heat. And then during division, they can get the doxo into the cells. And indeed, you have a percentage of dead cell increasing over time when you look at the cancer stem cells. So you have a way to treat not only highly proliferating, but also cancer stem cell. And when doing tumor initiating relapsing studies, so treating in vitro and then inject the in vivo on the flank of the animal, the cells, you look at the tumor-free animals, and we can see that indeed, again, over a very long time study, 250 days, only the group TR-doxo plus hyperthermia did not develop tumors, and they survive longer. Okay, we have also another system that has been studied in vivo, which is based on cobalt ferrite. These particles do not eat much, but what they do, because they have a high anisotropy, we have seen that at the tumor, they form very long chains. And these chains are providing mechanical stress together with the leakage of cobalt that is one of the components of their composition. And this, over time, produce, together with a very mild hyperthermia, produce a synergic effect that brings the cell to the death. Let me switch briefly to another concept. We use semiconductor nanoparticles in this case as radiotherapy materials. So differently from the others, we exploit cation exchange reaction. You have a semiconductor, you keep the anions, and you exchange the cations of the nanoparticles with a radioisotope. In our case, we have been working with radionuclide copper-64. And then we do this reaction, okay, either by cation exchange or intercalation. But the main concept is by going with the volume of the particles, you can exchange a large dose of radio isotope in a very tiny amount of nanoparticles. So you can have a very high specificity, radius specificity. And this was the type of reaction that we have set either in solution or in matrix. In solution, the idea is to have nanoparticles already in water, already stabilized with the linker, and you do the reaction to exchange. Okay, and this was done on zinc selenide, copper iron sulfide, and zinc sulfide with copper-64, and using the clinical available copper-64 solution. And of course, you can see that they have a very high radiochemical yield. And in addition to that, some of these particles, they have, beside introducing the radioisotope, they can heat up. So even after exchanging. And because of that, you can also use photo irradiation to provoke, let's say, the cell death. We have also combined magnetic hyperthermia with heterostructure that can do copper-64 exchange. One is iron gold copper sulfide, and another one is nano chains with zinc sulfide on the surface. These are the findings I wish to thank, and of course the group member. And thank you for your attention.
Thank you so much, Teresa. Very exciting, very nice. But because of the time, we have only time for one question. Anyone?
Was it too fast?
No.
Too many things together.
Oh, Alberto.
Okay.
Hi, very nice talk, Teresa. Just a very quick question. So I don't know if I lost the part of the presentation. The audio here is not working really well, but did you check, for example, for the internalization, the intracellular phase of these particles? If these particles are being accumulated, for example, in lysosomes or the parts of the cell. So this is the first question. The second question is more like related with the characterization of these particles. Did you check, for example, the possibility of dissolution of these particles, or even like the production of ROS, for example, from these particles?
Yeah, okay. Of course, I was first speaking about magnetic nanoparticles. So usually when you injected the tumor intratumorally, of course, they can spread and they can go, let's say, you can find them outside the tumor, but also inside the tumor cells. And usually they are, you increase the diffusiveness. So you find them in a sub-compartment like endosomes, no? Usually. The drug, of course, can be released either outside or inside. So you have the drug molecules that is either associated to the nanoparticles that has entered the cell through the endosome, but also because the process occurs at the tumor, you can have the drug exiting the particles before. So the diffusion of the drug is also through cell membrane. So in some cases we see both, no? This is for nanoparticles, the TR cubes. The uptake pathway of the semiconductor particles is the same, no? It's through the endocytosis, because they are sort of tiny. They are 20 nanometers particles. Usually they are well stabilized, so we care to have very single particles. So using PEG as a molecule to increase their stability in cell media, avoid protein opsonization. Also, we do that to ensure a single particle. So when we do TEM on cells, we can clearly see single cubes or single particles in cells. In some of the papers, you can find those images.
Excellent. Thank you so much, Larissa, for the presentation, for the answers. So we will now move to our final presentation, which is from the University of Sydney, and presenter Dr. Anna Waterhouse, and she will be talking about vascular nanomaterials. Anna, over to you.
Thank you, Wojciech.
And I'll pause. I know that you will be presenting some data which are sensitive, so I'll just pause the recording now.